Okay. So, right. So week 3. So what we went over for week 3, we should have gone over chapter 6, which is our genetics, the genetic engineering stuff, which will tie in very closely with the genetics. If you didn't get chapter 6, you're going to have a hard time with chapter 8. And we have chapters 9 and 25. These are nutrition and environment. And then we have chapter 10, which is our enzymes and metabolism. Chapter 6 and 10 tend to be the most difficult chapters for this unit. So I am going to try to spend a little more time on those than I am on 9 and 25. Okay, first things first with chapter 6. For our major concepts here, we talked about what the genome was versus what a chromosome is. A genome is all of the genetic information in a cell. Whereas a chromosome, they're like just structural units of that genetic information. And so in eukaryotes like us, we tend to have linear chromosomes, whereas eukaryotes tend to have circular chromosomes. Also, eukaryotes with their circular chromosomes, they seem to just have one circular chromosome, whereas eukaryotes have multiple linear chromosomes. So there's a big difference there. Distinguishing between genotype and phenotype. This is a thing that some people have a little bit of issue with. Your actual genetic code and what genes you have in your cells, that's your genotype. But which ones are expressed and how that presents in the organism, whatever you see physically or whatever you see physiologically, that's your phenotype. Physical, physiologically, pheno. Fa. So just think of it that way. But your genotype is literally your genes. So you might have a gene for blonde hair from your mom and brown hair from your dad, but you have brown hair. So your phenotype is having brown hair, but your genotype would be one blonde gene and one brown gene. Okay, so that's the difference. So that gets us into talking about the DNA structure, which DNA stands for deoxyribonucleic acid. And so it's the deoxyribo is the D, the nucleic Na acid. These is basically what our chromosomes and our genetics are made up of. Anything that is a cell has DNA for its genome. We already talked about the viruses can have DNA or RNA, but if you're a cell, you have DNA for your genome, and it's always just double-stranded, double helix, regular old DNA. The nucleotides that make up DNA, all nucleotides in general, a nucleotide, this is important to remember this. I know we learned about it in probably if you guys took GOP, or GOB, whatever it is, GOB chemistry, but we're going to make sure that we have that reviewed. So a nucleotide is going to be basically your sugar. This is always a 5-carbon sugar, and it's based off of ribose every time. It's based off of ribose. It's just whether you have an oxygen removed or not. So if you're deoxyribose, you just had an OH group removed. It's just an H now. And so that's the difference. It's just based off of this 5-carbon sugar ribose. And we have the phosphate, which we've already learned about. Phosphate is a phosphorus atom with oxygens all around it. And those oxygens are very electronegative. Some have double bonds, will have like negative charges on them. Very negatively charged aspect to it. And this will create these sugar phosphate creates the backbone of our DNA structure. And then shooting off of the sugar, we will have the nitrogenous base. Don't get confused by it being a long word. It's nitrogenous because it has nitrogen in it. And that's really all we need to know from my class. For my class, I'm not going to ask you about the purines or the pyrimidines. I'm not going to ask you about that because I get confused between them all the time. I have a freaking biochem degree and I get confused. So I'm not going to ask you about purine versus pyrimidine, but you do need to know what the DNA bases are. And so ATCG, it'll get you partial credit if I ask you a question about them. But ATCG stand for adenine, thymine, cytosine, and guanine. So in DNA, we have thymine. But if you were RNA, we would replace that with uracil. It serves the same function, but just in our RNA. All right, so that's the difference. Having deoxyribose instead of just ribose, we removed literally an OH group. We have a phosphate group. That's the same between DNA and RNA. And then adenine, thymine, cytosine, guanine, or if you're RNA, it would be adenine, uracil, cytosine, guanine. And that's really it. DNA tends to be double-stranded and RNA tends to be single-stranded. All right, because DNA is double-stranded, because DNA is double-stranded, and we would draw it like this, and you have your double helix and it twists on itself and all that lovely stuff, our DNA ladder. Because we have this backbone here, the backbone of the ladder, not the rungs, but the backbone of it, those are our sugar phosphate alternating. And then the rungs, these are our nitrogenous bases. And A on one side always binds with T on the other, and G on one side always binds with C, and vice versa. And so that's how one strand is made up. So if one strand reads A, C, C, G, T, T, A, the other strand would be T, G, G, C, A, T in your ladder. And then these these hydrogen bonds between these nitrogenous bases are what make up the rungs of our ladder. It's just hydrogen bonds, which is wild to think about, but it is what it is. And so that is the DNA double strand. The backbones run in opposite directions. So if you had sugar phosphate on one strand, then on the other side, it might be sugar phosphate, and whatever sugar phosphate. But think of them as being oriented differently. And this has to deal with how the actual structure of that 5-carbon sugar is set up and like which carbon. And if you guys learn in chemistry, we'll number the carbons based on like what they're bonded to and whatever. So we could number them literally like 1, 2, 3, 4, 5, whatever. And so when we talk about 5' to 3', it's literally just based on which carbon of the sugar, which I will never have you guys show me on a picture. I will not have pictures of it on my test. But being aware of that concept, it's just a matter of the sugar and how the sugar is oriented in the backbone. And so one side of the backbone runs in one direction and the other side of the backbone runs in the other direction, just based on how those sugars are oriented in the backbone. And so that's what makes DNA anti-parallel, that the sugar phosphate backbone runs in opposite directions for each strand compared to one another. And they're literally, we call it 5' to 3' because of which one is at the end at each side. That's it. So which carbon is at the very end on each side. And most of our processes that are going to happen in the cell will happen in the 5' to 3' direction, which we do need to remember. So that is something that you guys will get tested on, is knowing that 5' to 3', that's where normal cellular processes are going to happen. There aren't going to be any circumstances in my course where anything will happen in the 3' to 5' direction. It's always going to be 5 to 3. 5 to 3. All right. We talked about the hydrogen bonds. So anytime you're making a copy of your DNA, that is called replication. So replication is always DNA to DNA, always. Replication is DNA to DNA. And because the backbone isn't just a straight ladder, it doesn't just look like this, it's twisted a little bit, we have to unwind it first. And so the enzyme that helps with that unwinding is called helix enzyme. So that's the thing that helps with the unwinding. And then we separate those hydrogen bonds from one another and just open up that ladder. So after helicase kind of unwinds, then we have the separating and opening of the strands. So this might be the ladder, but then this side will be going this way, and this side will be going this way. And if this was A, that one would have been T. If this was C, this would have been G, and etc. All right, so we're opening up that double helix. And that site where we're opening is called the replication fork. Oh my gosh. Replication fork is where we are opening. One of the strands, because of the 2 strands that we've opened up now, one of them is oriented 5' to 3', So the copy that we would make would be going 3' to 5'. That's not going to work. Everything happens 5' to 3'. So one of the strands that opens up, that was the opposite way. When we're adding the new nucleotides, we can add it 5' to 3' just straight through. That's our leading strand. So that's the one that's going to be synthesized continuously. That's usually not too difficult for people to remember. The leading strand is the one that's made continuously in the 5' to 3' direction. The other strand, as it opens up, we will go from what is open in the 5' to 3'. So it's like you had your thing that was closed. Let me get an eraser here and make this bigger. Why? What is happening? Okay, hold on. I'm trying to zoom out and it's not. There we go. Okay. So let's do it over here. We have a lot more space over here. This was your ladder after helicase unwound it. And then we open it up with our replication fork. Like lungs kind of at first, but this is 3 and this is 5. And we can add nucleotides on this other side to match up with each of these in the 5 to 3 direction, going on into 3, and just keep opening that up and just keep adding and adding and adding. That'll be our leading strand. As we open up this one, which was 3 to 5, we can't add in this direction. It's not going to work. So what we can do, this is a little off from my pen, so just give me a second. So what we can do is, as it opens up, add in the 5 to 3 direction. And so this is the lagging strand, the one that's made in fragments. And those fragments are called Okazaki fragments. And you guys, you don't have to stick around, by the way, because I'm going to keep doing this if you're here or not. So, but yeah, you're welcome to move. But if you guys do end up needing to leave, because you know this one's probably going to go a little longer because it's a lot. So feel free if you do need to leave, because I'll keep going and record everything, just so you guys know, so you don't feel obligated to stay. But anyway, so we've got our leading and our lagging strand going. The fragments that we make as a result of having to do the lagging strand in parts are called our Okazaki fragments. And this is all just DNA replication. This only applies to DNA replication, this. All right. And then those fragments, because they are made in fragments, and this other one would have been made previously in another fragment if you counted it in, we will seal those together with the enzyme DNA ligase. It ligates. And so if you don't know what ligate means, and it is something that just means to join together. I used to play clarinet and saxophone. So they were woodwind reed instruments. And so we had to put a ligature on there, and the ligature joined the reed to the mouthpiece. So that's how I remember what ligate means, but it's the same meaning. So it's just joining it together. So DNA replication, that's that. So next is dealing with RNA. So RNA is ribonucleic acid. The DNA deoxyribo had an oxygen removed from the ribose. It's the same sugar. Ribose just still has the oxygen. It's not been deoxygened. So RNA serves several functions. The main ones that you need to know from my exam will be mRNA, tRNA, and rRNA. So let me get rid of this drawing that I have over here. We drew it. It was there. Come on, dude. I'm having such a hard time with this eraser. The eraser is the worst part of this. Just want to free up some space. Okay. We made our Okazaki fragments. Cool. Now we're on RNA. So the main ones that you need to know for my class are mRNA, which is messenger RNA, tRNA, which is transfer RNA, and rRNA, which is ribosomal RNA. And it serves as the structure of the ribosomes. And the process, we already said that DNA to DNA is replication. We're literally just replicating the same DNA molecule. But if we are going from DNA to RNA, they're both nucleic acids, right? But they're just different. So I think of this as the same thing as like when you guys are copying down notes of something that I've written down, then you're transcribing my notes into your writing, right? So that's what transcription is. We're still writing script, scribe, write, we're still writing, transcribe, and it's still in English. It's still the same language. It's still in the nucleic acid language, but we're not replicating. It's not the same exact molecule, especially because we're using uracil and we've got a different sugar now. It's ribose, but it's still nucleic acid language. It's just written in a different way. And so that's what transcription is. And this is just the process of taking the DNA template and turning it into the RNA. All RNA comes from our DNA. And so even though we think of our genes as coding for proteins, and that's what most of our genes will do, the other genes that are in our body code for the RNA that help build those proteins. So you have genes that are going to code for the tRNA, and it just makes the RNA, and that's the end product. Or genes that code for ribosomal RNA, and it just makes that rRNA, and that's the end product. But anything that codes for mRNA, messenger RNA, messenger RNA is just a working copy of a gene that's going to code usually for a protein. So messenger RNA is just the messenger of whatever that gene is. It's a working copy. The tools that we're going to use to turn that working copy of the gene into the protein product, that's the tRNA and the ribosomal RNA, rRNA. Okay. All right. And we already mentioned uracil. Okay, so this process is called transcription. Making that mRNA working copy is like our primary focus. I'm not as concerned about how the tRNA and rRNA are made, but they're also coded by your DNA, just to be clear. But going to get a protein out of your DNA code, we have to make a working copy of it. So I always imagine this, and one thing that I talk about in my lectures is, think of it as like you have your main instructions for how to build a protein in your Smithsonian institution. We have the bank that codes for everything that you could ever want to build in the Smithsonian Institution. You want to build a chair, then you might go to the Smithsonian Institution to get instructions on how to build a chair. But are you going to build the chair in the Smithsonian Institution? Probably not. And do you need to have the instructions for everything open when you're at home? Probably not. You're just going to make the instructions for the chair. So you go to the Smithsonian Institution, you write down instructions on how to build a chair. That's the transcribing part. Then you take those instructions home and then build a chair from those instructions. But the chair itself the same language as your written instructions or as no? So something else completely different, right? It's the same thing with the proteins. So we've made our working copy of just the gene that we need in our mRNA. Of your 20,000 genes that you have in your cell, we've made our one copy of what we need to make. So that's our mRNA. And this process of making that mRNA, going from DNA to mRNA, is called transcription. And it only happens in just 3 steps. The first step is initiation. Easy enough. So where are we starting this? How does initiation start? It's going to start always at the same place, at a place on the DNA called the promoter. Promotes the transcription. And so RNA polymerase, which is very similar to DNA polymerase, but we're making RNA now. RNA polymerase is an enzyme that will add RNA nucleotides, so a UCG, in the 5' to 3' direction, just exactly like how we were doing with our DNA replication, but we only need to make one strand. And so that'll happen in the 5' to 3' direction through our next step, which is the elongation. Add our RNA nucleotides. 5' to 3' direction. All right, so we bound our RNA polymerase at the promoter, and then we just start adding nucleotides in the 5' to 3' direction, building our single strand using the DNA as the template. So if there was a G on the DNA, we're going to add a C on our RNA. If there's an A on the DNA, we're going to add a U on the RNA, and so on and so forth. So complementary nucleotides using the DNA as the template. The next step is just the termination step. It will eventually get to a sequence where it knows it's done. I don't focus on that on the transcription side, but it's going to be the same 3 steps for translation, which is the next part. So translation termination is a little more important, but it'll get to a point where it stops and we're done making the mRNA, and that's transcription. And that's it. We've made a copy of our DNA gene. Then what happens? We've made our mRNA. And if we're building our mRNA, according to the template on the DNA in eukaryotic cells lives in the nucleus. It's in a whole membrane-bound structure, right? It's a whole structure inside of the cell. And so you make your mRNA. Transcription occurs in the nucleus for eukaryotes. But bacteria don't have a nucleus, right? They're eukaryotes or prokaryotes. So you can technically transcribe all of this, and then a ribosome can bind onto it and start translating while transcription is still occurring, which is very advantageous for bacteria, and why they can actually multiply so much faster is they don't have all these constraints. Now we need the constraints in our cells because we have a lot more going on in our cells. But for them, that lets that process be a lot better. This is actually kind of fascinating watching them clean that window out there. That huge pole is wild. Anyway, so that's transcription. The problem with eukaryotes, also because we're so complicated, we also have extra bits in our DNA. So after you have made, so this all leads to our mRNA transcript. But our problem with eukaryotes I'm so sorry. Someone just someone left their water bottle out here. Okay, so the problem with eukaryotes is that the mRNA transcript has extra bits in it that we don't need to read, or they might mean something for this transcript, but not for another, or something like that. So we will need to edit it down. So eukaryotes go through a process of editing called splicing. We need our exons. Exons are the important ones, and we will get rid of introns and just splice together all of the exons. And then that will make the readable transcript. Now, this is important to remember because when we get into chapter 8, we won't be able to understand if you're a bacteria and I just put eukaryote DNA into the bacteria can't splice the exons together. And so you have to like take a workable, readable mRNA that's already been spliced and turn it back into DNA through a process of reverse transcription, which we have HIV to thank for that discovery. So now we're able to basically put human genes that have been processed, they've been transcribed, the mRNA has been spliced, and then turn it back into DNA and put that already processed DNA version into bacteria. And now bacteria can make insulin for us. They can make other human products for us, and we can use that to our advantage. So that splicing is just something that happens in our cells, but it directly affects the genetic engineering chapter that we get into next. Okay. Hold on, I'm just going to email this person back real quick. I'll leave it in my office for you. Okay, anyways, so that's where the introns and the exons come in. So we get spliced out and the exons we keep. I don't have a great way of remembering which one is which for this one because I get confused because I'm like, the introns stay in is how I want to think, but that's not correct. The exons are the ones we want to keep. The introns, we're going to cut them out. Okay. All right. When you look at, I'm sure somebody else has a better explanation than that, but that's my, it just is what it is. All right, so next is going into translation. So I'm going to get rid of this stuff. We had it for a little bit. It was nice while we had it, but I want space. All righty. So we already said DNA to RNA. With transcription, it is the same language nucleotide-wise. But now we're going to translate it in a totally different language. And so that is going to take us from our RNA to our amino acid sequence, which will be our protein at the end. And this is translation. So now we're no longer talking about nucleic acid language. We're talking about amino acid language. It's a different thing altogether. So this is actually translating it to a different language. And so the process of translation occurs in the same steps. There'll be initiation first. Initiation, we will find a start codon, which is always AUG. So codon is a 3 nucleotide sequence on the mRNA transcript, and it stands for something. The 3 nucleotides will code either for an amino acid or tell us to stop translating. That's the only possibility. So the start codon is always AUG, and it's always going to result in the same amino acid, which is methionine. For my course, you do not need to memorize which amino acids. You don't even, the structure of the amino acids, don't care. The names of them, you don't need to memorize them. The codon, which one codes for what, don't need to know it from my class. If you're taking biochem, yes, you need to know that stuff. But for me, I just need to know the concepts behind this stuff, okay? So AUG is the only important one for the codons that actually code for amino acids. And then the other ones, they might just be other things like GGA or UUA or something else, and they'll code for other amino acids and whatever. So those are triplet nucleotides on the mRNA that code for either an amino acid or a stop, and those are the codons. How do we get from a codon to the actual protein result? Well, what happens is after we start at the start codon, this is where the ribosome binds. And then the next part is the tRNA that has the anticodon that matches the mRNA codon, carries an amino acid with it. What it looks like is you might have your mRNA. I'll just put an M here so you know what that is. That has AUG. Okay. And then your tRNA comes in and it looks like a squirrely little T. And give myself a lot of space, but whatever. Then this would be UAC. And that this up here on the tRNA is our anticodon. And this is our codon. And attached to that tRNA, you have the amino acid. In this case, it's methionine, MET. And then the next one would come in, and that would be, so this is just our initiation right here. And this happens inside of the ribosome structure. And the next one would come in our elongation. So which is our next step, elongation, which is very similar in concept-wise. So instead of adding nucleotides, now we're adding amino acids according to the codon sequence. So I don't know my codons anymore, so you're just going to have to bear with me making things up. So this will be like, I don't even know, AAA. So our anticodon would be UUU. I can at least tell you that. UUU. And it'll be attached to, I don't even know what, like, let's just say phenylalanine. And then the methionine will create a bond right here. And that bond between the methionine and the phenylalanine, whatever your next amino acid is our peptide bond. And this will continue on and on and on until we get to a stop codon. There's 3 different stop codons, and you don't need to memorize them either, but there just are 3 of them. In this case, one of them might be UGA, and this doesn't match. There's no tRNA that matches with that. And so the ribosome says, there's no tRNA that can bind here, we're done. And it just pops off. So this is our stop. This is one of the stop codons that cause that to happen. And because these codons don't have a tRNA match and they don't have an associated amino acid, we call them nonsense codons. And so that is our termination step. And whatever you get with your methionine linked to your lysine, linked to your phenylalanine, linked to your isoleucine, linked to your glutamate, whatever your amino acid sequence is, just to remind you guys, that'll be your primary structure, right, of your polypeptide. Polypeptide is exactly what it sounds like. It's just multiple peptide linked amino acids, and that's it. We might make other modifications to our polypeptide. Then it might associate with its neighbors, causing our secondary structures, our alpha helices and beta sheets. Then our tertiary structures can form where we have binding across the whole polypeptide and create our bow. And then that polypeptide might work with another bow polypeptide and create a functional enzyme. And then that would be the quaternary structure of our protein. But that's where we get from our DNA to our RNA through transcription to make those components. And then taking that RNA and then reading it and creating a protein out of that. And that's what these processes are. So this replication is DNA to DNA, transcription is DNA to RNA, and translation is RNA to protein. And this is our central dogma of biology and how cells work, is that they have to go through this process. All right, we following so far? At least the general aspects of it, I hope. Okay. So now we're getting into the operons, and people find these tricky too. So I'm going to try my best here. I feel like I just talked about the operons, but it's just because every semester I talk about the same thing, and so it always feels like that. Okay. So the other thing that our eukaryotes have is something called operons. So eukaryotes don't have these. It's only our prokaryotes, eukaryotes, whatever you want to call them. And primarily, we're just talking about bacteria. And these are called operons. Operons are just a set of genes that are going to be controlled by the same promoter. It's one promoter that whenever our RNA polymerase binds to that promoter in the DNA and starts for transcription, it'll transcribe a whole bunch of genes all at once. And so they're all linked together. And they're all controlled by something next to the promoter called an operator. And they all work the same, always. So it works like this. You will have your DNA sequence, whatever it is, and you'll be getting ready to make your RNA. And on your DNA, you will have your promoter region. This is where your RNA polymerase will bind and start adding the nucleotides for the RNA. Next to that promoter region, we have an operator. And then after that, you have your genes, gene 1, gene 2, etc. Whatever going on. Okay. This is all on DNA. We're just trying to make the RNA. And so what happens is that RNA polymerase, which I'll put RNA pol here, would normally bind onto that promoter region and just squeak on through. But what happens with this is that we are actually able to bind a little guy onto our operator region, which I'll put an R here, because the R, we call it a repressor. So when the repressor binds to the operator, RNA polymerase can't move from that promoter region. It's just blocked. It's a physical blockage. So repressors bind to operators so that the RNA polymerase can't transcribe. That's how all operons work. Whatever kind you are, that's how it works. So then it's just a matter of, did you start this way or were you working and then you got stopped? And that's the difference. It's the only difference. And so in this, if it started this way, if it starts off, it started with a repressor there. That's why it was off. Repressor was already there. So ones that we start that are off, if your patient isn't going into labor, but they should be, and you want to make them go into labor, what's the process that we call that? Inducing. So we can induce this to give birth to our transcripts, okay? So we can induce those. The ones that start off are inducible. If you didn't have the repressor there and you were able to work and you started on, then you're repressible. We can repress you. That's it. Don't read too much into it. The lack operon that it talks about in our lecture, in our textbook, in our notes and whatever, is one that starts off. And what happens is when lactose, the sugar, there's just lactose, like in our milk, is present, then the bacteria what happens in the bacteria is the lactose binds to the repressor. And when that happens, the repressor is a protein. When it binds to lactose, it changes shape and goes away. And now we can read through. That's what happens in the lac operon. When lactose is present, then it binds to the repressor dissociates, and now RNA polymerase can do its job and make the genes. The genes that are downstream are there to break down lactose. That way, we're not making the genes to break down lactose unless lactose is present. Why would we make the genes? Why would we waste our energy and our resources making those genes when we don't need them? So that turns on making those genes. It is an inducible operon. The example that it gives in the book for the repressible operon would be the arginine operon. Basically, what happens is arginine is an amino acid that you need in your body just to build proteins, right? So let's say you're having an off day. You don't need to build as many proteins. So you would build up a whole lot of excess arginine. You're not using it up. The cell's not using it up. So what happens is if you get excess arginine, it's like looking in your warehouse and saying, oh, we were making dresses. People aren't buying the dresses. And you have a whole bunch piling up. People just aren't buying them anymore. Maybe it's wintertime. And you would say in your warehouse, oh, we need to stop making dresses. Well, your cell says the same thing. We need to stop making arginine. So what happens is when you have dresses lying about, the dresses would basically say, whoever the floor manager is, say, we have extra dresses, stop production. If you have extra arginine, the arginine binds to a repressor and causes it to bind on to the operator. So the arginine binds to just the repressor floating around in the body. So I say body, the cell, the bacteria. And that bacteria, that repressor picks up the dress, picks up the arginine, and says, oh, we've got too much of this. We're building back up. We need to stop making arginine. And so all those genes that were involved in making arginine, we'll turn them off. It's just a way of signaling it. And so that's all that's happening in those. So don't read too much into repressible and inducible operons. They're literally just either inducible, we can turn them on because they were off, because the repressor was blocking. We can remove that now and turn it back on. If you are repressible, you are on, and now we can repress you by making the repressor bind to the operator, blocking RNA polymerase. And that's all that's happening. Usually repressible operons, like the arginine, it's usually the product, whatever the ultimate product is. So these several genes are involved in making arginine, but whatever the ultimate product is, the arginine, usually if it builds up, that causes the repressible operons to be turned off. That's how we repress them. It's usually the product of that. Okay. I don't know if that helps at all with I don't know if you guys have read it all into that chapter or like looked into that, but a lot of people have a lot of confusion with those. Focus on the words inducible and repressible. Okay. So if it's inducible, were you on? No, we need to turn you on. You know, well, if your patient isn't in labor, but they need to be, you're going to turn that on for them with your drugs. So just start there. That's how I recommend. Okay, so we did that one. The other thing is horizontal gene transfer. There are 3 types of ways that bacteria can exchange information that isn't hereditary from the parent bacteria. And so bacteria can actually exchange information with other bacteria through horizontal gene transfer. So horizontal versus vertical, this is very actually important in genetics overall. Horizontal just means that we're exchanging genes between non-parental methods. Vertical transmission or vertical, anytime we talk about vertical, it's parent to child. So, you know, parent to offspring, however you want to look at it. But like if you were a bacteria and you divided and you had antibiotic resistance and now your 2 daughter cells have antibiotic resistance, that's vertical. But if you are a E. coli and you come up to another E. coli and that E. coli had antibiotic resistance and can shuttle it to you somehow, that's horizontal. Okay. So it's it's just not parental. So this is horizontal gene transfer. There are 3 types in bacteria that are important for gene transfer. The ones that we're worried about: conjugation, just like we think of conjugal visits in like a prison or something like that. Conjugation, think of sex. Okay, so this happens through the sex pilus, and we learned about the pili, which is multiple pilus, in chapter 4. So this is a channel that some bacteria, it's usually gram-negatives, can form between their cells, and they can just shuttle DNA through them. And they can make whole copies of whole plasmids. And we learned plasmids are small extracircular bits of DNA that aren't essential for living, but can help survival. Things like antibiotic resistance or even toxin production or something like that happen with the plasmids. So that method of shuttling it through a channel that is like bridging between two cells, that's the sex pilus. That process is conjugation. The next one is transformation. Transformation is an experiment that we'll be doing next week, I think. It's exercise 10. And so lab exercise 10, we're going to be doing a simulation of transformation. It's really not difficult, but all it is that some bacteria can pick up DNA that's outside of their cells. They have like pores that enable them to like pick it up from outside. And that's all it is. So they are transforming themselves with that new DNA. Cells that can do that, that can pick up exogenous outside DNA, we call those cells competent. So that's another term to be aware of. Competent cells pick up outside DNA, call that exogenous. All right, the third one is called transduction, which we actually have already talked about a little bit. We just didn't call it transduction. So transduction is whenever you get new genes from being infected by a phage virus. So when a phage infects a bacteria and inserts its genes into that bacteria, and that bacteria gets new traits from those genes, we call that bacteria, it's been lysogenically converted, right? We learned about lysogenic conversion. The process by which all of that happened, we call transduction. It's a method of horizontal gene transfer. This happens because some phages will come, put their DNA into the bacteria DNA, and when they come back out again to make new viruses, they take some bacterial genes with them. Sometimes that's predictable and sometimes it's random, just depends on the virus. But they can actually take genes from that bacteria with them and put them in the new offspring. And then that's how they get inserted into the next cells. So that's transduction. So conjugation, sex pilus. Transformation, pick up outside DNA. That competent cells can do that. And then the third one is transduction. That's the phage lysogenic conversion thing. All right. Then we have the different kinds of DNA mutations that the chapter talked about. There's major ones that I just want to cover briefly. Come on, work with me here. There we go. Clean this up. Operons. That was fun. All right. All right. So our mutations. I think there was like 5 group major ones that I want you guys to know. Might be just be 4, so don't hold me to 5, but I think it's 5. Yeah, it has to be. Okay. The first one is missense. It doesn't matter what order you remember these in, but missense. Missense is whenever you have a codon that might be like AAA that codes for phenylalanine, and that AAA gets changed to AAG, and now that codes for, I have no idea, glutamate. It's a different amino acid. Second one would be silent. We're gonna have the same amino acid, but a different nucleotide sequence. So if it was AAA, and that got changed to put GG on that, but that's whatever, who cares? AAA got changed to AAC, but they both still code for phenylalanine. It's a different nucleotide sequence. You definitely changed the nucleotides, but it still codes for the same amino acid. Now, this is actually pretty common. Most amino acids are coded for by more than one codon. And so this is what we call the wobble effect. That usually happens in the third nucleotide of a codon that we see the difference, but they still code for the same amino acids. Some amino acids are coded for by 4 different codons. Some of them are only coded for by 2, and there's even some that are only coded for by 1. Like AUG is always, like methionine is always coded by AUG. You can't have multiples of it. So when you have this effect, that's the silent mutation. The third one is going to be the nonsense. This is whenever you have a stop codon instead. So instead of AAA, maybe you changed and you coded for phenylalanine. Now you have gone to UAA, I don't know. And that's a stop codon. It's a nonsense codon. It doesn't code for an amino acid. So that's why it's called nonsense mutation. There's also back mutations. So let's say that a strain of bacteria, we see some of the progeny that had this missense mutation. But now they're going through another mutation where they're going back from AAG to AAA. That would be a back mutation. So if we consider the wild type, what we expect the gene to read, and somebody had a deviation at some point, but now they're going back to the wild type in the mutation, that's called a back mutation. So reverting to what we call wild type, and that's just what your expected genetic sequence would be. And then we have frame shift mutations, which are sometimes difficult to think about, but they shouldn't be. So the way that I think about it is like reading a book. So if your book said the cat sat, I don't even know, it doesn't matter. The cat sat. Let's just say that. Okay. And there's more words after that. The book continues on and on and on and on. So a frame shift mutation is where you delete or add a nucleotide, but you still have to read in 3 frame. So instead of it, the cat sat, now you have the cas at whatever, blah, blah, blah. And so now you have changed your nucleotide reading frame completely because it always happens in 3s. And so everything downstream from whatever your frame shift mutation was will be affected from that. And so this can be actually catastrophic for protein production, as can the stop codons and stuff like that. Those are usually the worst ones, will be the frame shift and the nonsense mutations. Okay. Chapter 8. Whew. All right. I'm trying to cover the content now so that when we get to the questions, we can just do the questions. All right, so chapter 8. So this is where we're getting into our tools for genetic engineering. So in chapter 8, we introduced the restriction enzymes. These are just proteins that are enzymes in bacteria naturally that bacteria use to cut out foreign DNA. But they create these sticky ends and they're always at palindrome sequences. So if it was GCC, it would be CCG. So first of all, it reads with your codon separation, GCC, CCG. But then your opposite for it would be CGG, GGC. And so it would read the same forwards and backwards on the strand and be complementary with each other. So this is what we call palindrome. I'm messing it up. I know I am. Hold on, let me fix it. I always do the same one, and so I need to stick with the one that I always do. Oh my God. Erase and not next slide, please. Okay, thank God. All right, I need to stick with the GAATTC one. All right, so when you have GAATTC on your DNA, the opposite strand of your DNA would be CTT. This is it. This is the better one. Just a better example. So this was your DNA double strand, right? So this GAATTC, but you read this one in the opposite direction. They read the same in the opposite direction. That's why they're palindromes. And so then also, if you were to fold this back on itself, they would match to each other. This is our palindrome. This is a way better one. Try not to make them up on the fly anymore. They read the same backwards and forwards on the strands, and they could also technically, one strand could fold back on itself. So that's the palindrome. And the way that they create sticky ends is that the enzyme would cut like here. And then your resulting would look like this. You'd have on your double-stranded DNA, you would have G on one side and CTTA, that'd be it. And that would be open. There'd be nothing on the other side. You would have 2 ends. And so the other side would look like AATTC and have a G. And then you can see how this sticky end would want to match up with this other sticky end. Right. So in regular old DNA, when you're cutting regular old DNA, those ends are going to want to match back up with each other. just because of the complementary A to T, C to G, B, S. So, but the restriction enzymes, what they'll do is allow us to cut there. And then you would have your other sequence, like let's say you cut bacteria DNA with this enzyme, but you also have human DNA that you've removed your introns and exons and turned it back into DNA, what we call, cloned DNA then, and take that DNA and cut it with the same enzyme, and it's got the same sticky ends. Well, that little gene for insulin that you just made, now you can insert it into the bacterial DNA with those same sticky ends, and you'll give your gene in excess. So that's more likely to bind with your gene than it is with its other end. And so that's how we would favor inserting genes. And this is a extremely common tool used in genetic engineering. And it's pretty much how we do all of our genetic hybrids by hand. And so when we talk about GMOs, most of our organisms that are genetically modified organisms, which is what GMO means, transgenic, the same exact thing as GMO, they're done this way. And we engineer it to work that way so we can control those genes and how we're inserting it. When we look back in history on Gregor Mendel and how he used to like take plants and like make them interbreed with one another and then have different colored sweet peas and whatever it was that he had, that's still genetic engineering. They're still GMOs. GMOs have been around since before this has been around for a very long time. Now, if you read the chapter, then you probably know there are definitely pros and cons to GMOs. It's not all good. I'm not going to sit here and tell you that it is, but probably what you think is bad about a GMO is not what you think. So the likelihood of, if you make something this way, of those genes jumping out and affecting your genes, 0%. It's not going to happen. So if you eat a GMO product, those genes are not going to jump into your DNA. You're not at risk for that. But the risk is great whenever we look at the environment and how it's impacted. So whenever we are inserting genes into corn to make it resilient to pests, well, corn can interact with other plants. They can spread those genes through like pollination process and things like that. And now other plants are resistant to pests. But where are the pests going to get their food? And now you have the pests and the birds that rely on eating the pests. And so that's the bigger issue with GMOs, how they affect our environment. Not you. You're not going to get those genes into your body and whatever. The implications in animals that are GMO'd and whether that's ethical, that's a question too, right? So that's another thing. But don't be the person that thinks that I'm looking at you too, because you're the only ones here. I'm not saying you guys think this, but like anybody listening to this, don't think for a minute that if you eat a GMO product that you're at risk of your genes getting changed by it. That's not a real thing. But do be aware there are risks with GMOs. It's just not that. Okay. And that talks about that in this chapter. So it is actually relevant. I'm not just getting off on a soapbox here. Okay, so that's restriction enzymes. They're enzymes that cut at those palindromic sequences and create the sticky ends. And then we can use that to our advantage to do that. But in our bacteria that are doing that, they are only able to cut at that sequence. So there's one enzyme and it always cuts at that GAATTC, period. Then there's another enzyme that cuts at another sequence and another enzyme that only cuts at another sequence. And we haven't been able to manipulate it to cut at any sequence that we could want. But CRISPR-Cas9, we can do that with. So with CRISPR-Cas9 system, Cas9 is a set of genes that codes for enzymes that we can manipulate how and where they cut in the DNA and could potentially allow us to cut anywhere in anyone's DNA. And we are finally seeing major medical breakthroughs with CRISPR and Cas9 in living tissue, where we are able to take out somebody's T lymphocytes. Let's say somebody that has HIV, we can take their T lymphocytes and use CRISPR-Cas9 to treat their T lymphocytes and actually remove that HIV DNA from those T lymphocytes with CRISPR-Cas9 and then give them back their own T lymphocytes. And they're no longer infected with HIV. And this is showing to be something that is actually proving effective. So that's a theoretical situation. That's the hope of what we want it to do. But there has been plenty of situations now in human biology where we're actually finally getting CRISPR-Cas9 to show some results in genetic manipulation in living humans. Now, we're making changes to cells that are already developed. They are not stem cells anymore, and they're not their gametes, right? And so that's where the end of this chapter starts talking about gene therapy. So this relates to that. So I'm jumping to that right now. We already talked about GMOs, but let's talk really quick about gene therapy because we're talking about CRISPR-Cas9. With CRISPR, you can make these changes potentially to an embryo, like after it's been fertilized. And we are seeing now with CRISPR, they are doing experiments where they're actually using CRISPR to remove that extra X in trisomy 21, which we see with Down syndrome. So we can actually have embryos that have Down syndrome and use CRISPR-Cas9 to edit their genetic information. And then you can have a baby that doesn't have Down syndrome. And they're actually using it now to potentially even look into treating people who already have Down syndrome with that CRISPR-Cas9 and potentially treat. I know, it's so good, right? Like, it's wild. It opens up a door to things that are terrifying. But the idea of how it can actually help people that have these diseases that are seriously affected, but can you live a life and have a full, happy life with Down syndrome? Yes. Can you live a happier one without it? I mean, who's going to argue otherwise? So we can help that. And even diseases that are actually like genetically detrimental, that are actually going to be harmful to you having, whether it's Huntington's disease or something that you're guaranteed you're going to die young from. Maybe, things like X-linked SCID, which is the severe combined the blood boy in the bubble disease, stuff like that, where we could actually manipulate your genes after you've been born and then say, hey, you don't have this problem anymore, and you never will. Your genes are fixed. And imagine it. But then also imagine people are paying money so that their kid will be tall enough to be a basketball player or more beautiful or have this color of eye or this color of hair. And so if you guys have heard of the movie Gattaca, so it is who was in that movie? Ethan Hawke. It's like he recently became more of a villain character, but he used to be more of a hero character. Anyways, Ethan Hawke was in that movie. It's Uma Thurman. And the idea is that they're genetically engineered people, and they're meant to be engineered to do like certain jobs, or be more beautiful, be more effective, be healthier. And this guy comes along who was naturally born. He wasn't genetically modified, but he wants to go into space. And so he doesn't pass the checks genetically, but he's like, I can do this. I don't need to be, genetically modified to do this. And so he goes undercover and like uses somebody else's genetic identity and like leaves skin cells from the other guy and whatever. Scott, Jude Law in it is like the perfect guy or whatever that he's stealing the skin cells from, he's working with. But Jude Law had like an accident where he couldn't walk anymore. So he was paralyzed, had nothing to do with genetically. But anyways, it's a really cool movie, but it actually applies to that idea of what we call eugenics. And so it's the idea of trying to choose the perfect genetics, but then the people who are naturally born or whatever, or can't afford to have their genes upgraded or whatever, the ethics behind that is pretty serious. So the bioethics behind that. So CRISPR-Cas9 is great for treating diseases that need to be treated, but it also opens the door to designer babies and things like that, and the ethics of whether or not that's appropriate. So that's why I bring it up. And if you can't treat the gametes, or if you're not treating like the cells of before it's actually truly like a fetus, if it's just a implanted egg and a few cells, like you might be able to treat it there, and then that person could grow up, and then their babies, like their gametes that they have, their eggs or their sperm, will be fixed also. But if you don't treat it at the early stages there or before, like when it's just a sperm or an egg, if you don't treat it there, then it's not going to be inherited. And so, and most of this gene therapy is something that if we treat it in your body cells and they've already gone through development and everything, you can't pass it on to your offspring. We call that somatic gene therapy, whereas germline gene therapy is going to be gene therapy that actually affects heritability. And so the goal is we could eventually make all of these changes heritable and nobody would have any genetic diseases anymore. But there's also the other risk of the ethics behind everything else. So that's the gene therapy side. Okay, so we did CRISPR-Cas9. Other tools for genetic engineering: nucleic acid hybridization. If you were like, oh, this sequence of GTTCCA, whatever, this sequence is also always associated with breast cancer, let's say. So you could make a probe of this sequence and attach it to a fluorescent molecule, and then take your patient's breast cancer biopsy and mix up those cells with this probe. It's more complicated than that. You'd have to break them open and everything like that. But let's say we break open your patient's cells, expose their DNA, mix this probe with it. If we see that your patient's cells glow blue or whatever, that means that they had to have had that sequence and therefore they have breast cancer. So it's just matching the genetic sequence and seeing if you have that match in your DNA. So we don't have to sequence your whole DNA. We don't have to do PCR and multiply it out or anything. We can just use the probe to see, do they have a complementary sequence to this? And if they do, they have that gene. So this is used for genetic testing as well as diseases, because if there's genes that are associated with E. coli, I can take your patient's swab and then do nucleic acid hybridization with that. We learn more about this in chapter 18 with our diagnostic procedures. This is introducing just the concept of how it works. So that's nucleic acid hybridization, just seeing if they match sequence. In PCR, PCR stands for polymerase chain reaction. That's what it stands for. And that's, I feel like that's a question on the test. It's just knowing what it stands for. It might be like a short answer question. I don't 100% recall, so don't hold it against me if it's not on there. But it used to be. Polymerase chain reaction. I know for a fact you'll get asked about the steps in PCR, and there's only 3 major steps. I know that's on the test. So the 3 major steps in PCR, and it's in the notes and it's here as well. There's 3 major steps. The first step is denaturation. First step is denature. So taking the natural setup of your DNA, which is a double strand, and using heat, which I'm going to put this triangle. You guys know what that means. Yes, it's heat, delta. Heat. And then that will split up your strand. Just split the strands apart. This is denaturation. Using heat to separate your DNA strands. The second step of PCR is the anneal step. Anneal, primer, whatever you want to call it. But annealing just means to stick things together, sort of like ligating. Okay, similar concept here. So anneal. So what you're going to have are your separated strands. And then we will bind primers onto them on like one end. That's it. The primers bind. And DNA polymerase cannot add on to nucleotides unless there is something to build off from. And so we have to have primers for the DNA polymerase to even work from. And so these are usually pretty long. They're usually like 30 nucleotides. And that way you can target specific genes. So if your gene is there that you're targeting, if your patient has E. coli and they have E. coli genes there, then your primers will bind to the E. coli genes. But if your patient has salmonella and your primers only look for salmonella genes, or I of course messed that up. Of course I did. Let me think of this a different way because it's getting, I don't want to get weird with it. If your primers look for, let's just, whatever. Your primers look for COVID, because I feel like people know that we test for COVID with PCR. They know that we test for flu with PCR. Let's do that. So if your primers look for COVID, they're only going to bind with COVID genes. So if COVID is there, your primers will bind, and then your nucleotides can multiply from your DNA polymerase in the next step, which is your elongate step. Okay. But if your patient has COVID, flu, whatever, another thing, not what you're testing for. If you're testing for COVID and we're looking for COVID, it'll bind if your patient has it. If your patient has something else, flu, cold, RSV, right? There's no COVID gene for the primer to bind to, so it won't bind and you can't build off of it. So that's how PCR works. So it depends entirely on the primers. If your primer bound to the gene, you have the thing. And now we can build our nucleotides off of it. So that's the third step, is the elongation. Again, it's elongation. Don't we love it? Denature, anneal, elongate. And this is where we're going to have each step of PCR yielding 2. So each round of PCR, in round 1, we went from 1 DNA sample to 2. In round 2, we started with 2, which was our result from before. And it's going to become 4. It doubles every round. So it's an exponential doubling. So if I were to ask you guys, and I don't ask this on my lecture exam, but you'll get asked this on the lab exam, okay? Because we're going to talk about this stuff a little bit in lab too. If I were to ask you guys, if we went 4 rounds of PCR and you started with 1 strand of DNA, if you went 4 rounds of PCR, how many strands would you have at the end? You could walk it out like this, or you could look at it as an exponent as well. So if you start with 1 and you get 2, and it would be like however many rounds, and you're doubling every time. So it's like however many rounds, 2 to the whatever round it is. Okay. But you could also just walk it out, which is easy enough. So if you're at round 1, you went from 1 to 2. If you went out round 2, you went from 2 to 4. So what's round 3 going to be? 4 to 8. And then round 4 will be 8, 2, 16. Yeah, so it's really not that hard to walk it out. Just FYI. So that will be on the lab exam. We'll go over it again when we get there. But I just wanted to feed into that so that we could see how that would relate later.
Do you have a question?
Yes.
So like when the doctor, say we swab somebody in the ER for COVID and flu with the same swab, do you have to run 2 different PCRs, or is there a way to run the same test looking for both?
Right. So they have them designed now that we can do PCR for both now. That's a newer thing, and it works a little bit differently. They have primers for both flu and for COVID, but they have a way of knowing which one is which when they get their result. And so it's complicated on knowing how that works. I don't want to get into that stuff because it's just going to make it confusing. But yes, so they can actually do more than one now, but they're very specific, right? And so we have PCR now where you can like do one swab and pop it into one PCR tube, and it will test for flu, COVID, and RSV all at once. And so they can do that now. They've got it down enough to an art where there's some cases where they can do multiple at once, but they're very limited, and you only know, like, you know what you're testing for in that specific thing, right? Even like in food testing, whenever we were testing a lot of like salmonella and E. coli, like I had to run a different test for salmonella and a separate one for E. coli, a separate one for Shigella, a separate one for whatever, Listeria, whatever it was. So we had to do separate PCRs for every single one of them. And depending on how long the gene is, it might be a different amount of time, depending on how many A's and T's versus G's and C's. Some of those guys have 3 hydrogen bonds versus 2. So you might have to have higher heat or longer heating times. It's just like, so then the chemistry of that stuff makes it more complicated. But they've got those down now with flu, RSV, and COVID, where they can run them on the same cycle in the same tube, but even tell which one you're positive for at the end of it, which is pretty cool stuff. You can actually watch what we call real-time PCR and quantitative PCR. But like if you have your PCR machine going and you start adding nucleotides, each nucleotide, the A, the T, the C, or the G, each one is labeled with a different fluorescent molecule. And when the A gets bound on, it emits blue. When a T gets bound on, it emits red or whatever. And so the machine can actually track that and tell you what nucleotides are getting added on when. And then based on that pattern is how they can tell you whether it's flu or COVID or whatever. That can also tell you, like, if you're adding on nucleotides and we're getting flashes of light when you're doing that, and you're getting that consistently round after round after round, I don't even need to wait for it to finish, because I know that if you're adding on nucleotides, you have the gene. And so that makes that faster too, because a real life PCR doesn't take an hour. Real life PCR in like research and actual proper testing, it takes like 4 hours. But I can tell within an hour if it's going to be positive or not, because it's adding nucleotides. And so I don't usually have to wait after that. So yeah, so that's where PCR comes into play, but you have to have the primer. And so you have to know what genes you're looking for, and you have to know the gene. And so if your patient has a diarrhea disease and you've done all the normal testing for salmonella, for E. coli, rapid testing, whatever it is that you have, they're all negative. You can't just run PCR to figure it out, because you'd have to know, okay, what am I looking for? I need primers to look for that thing. And so you'd have to do other approaches to help you whittle down what it could be if it's not the ones we already had primers for. Does that make sense? So it could be process of elimination, but it also could be people come up with some weird stuff sometimes. They get the weird stuff. It might be rare to have the weird stuff, but some people get it. And so we don't always have PCR for those. So yeah, it doesn't just test for anything, right? It tests for the specific thing you're looking for. So this is the 3 steps. It's it. That's it. Denature, anneal, elongate. And then we will denature what you just elongated and then anneal it again. It's just those 3 steps over and over and over again. We'll do that usually anywhere from 30 to 100 times. So you'll start with 1 DNA, and you can do PCR from just 1 strand of DNA. We don't need very much. And then you will have billions of copies at the end of the whole thing. So it's pretty useful for genetic engineering, but also for identification of diseases that way. Because that person only needed to have 1 virus on that swab in order for us to do PCR. But would 1 virus pop positive on a home test? Probably not. It's just not sensitive enough for that. So yeah. Gel electrophoresis is another tool that we use for looking at DNA and separating it out. So I can take your DNA and I can take my DNA, and I can treat them with that same enzyme, restriction enzyme we were talking about before, the GAATTC. And I can cut it. I can cut your DNA with that, just all your DNA. Doesn't matter. I don't need to be specific. Just take your cells, open them up, get your DNA out, cut it with that. When I cut your DNA apart and then I cut my DNA apart, I can separate any of the resulting fragments out using gel electrophoresis. And then we can look at the bands on where they get stuck in the gel. So the bigger that you are, the more easily you get stuck. And so the smaller fragments travel faster and the bigger fragments get stuck. And so the smaller ones are going to be at the end or the bottom of the gel, and the big ones get stuck at the top of the gel. And then the thing that helps them even move through the gel at all is electricity. We already know DNA has a negative charge. It's got phosphates in it. Yeah, a lot of phosphates. Sugar phosphate repeating in the backbone. So DNA is very negatively charged. So it's going to want to move to the positive pole in that electricity movement. So the DNA migrates to the positive pole, but the large pieces get stuck and the small ones travel fast. And so you'll get these gels. You guys have seen these before, yes? So it looks like the gel looks like this, and you put your sample in here, and this would be like patient 1, and this would be patient 2, and you cut them up their DNA, and then patient 1 might look like this, and then patient 2 might look like this. Yeah. And so these are the smaller ones, and these are the large. I don't even need to freaking sequence your DNA. Nobody has the same footprint as another person, period, except for identical twins. That's the only time. And you can even see sometimes there's even differences there depending on what DNA you're testing and what part of the body and stuff like that, just because of epigenetics, which we're not going to get too much into. But when we start learning about immunology, your body is trained to adapt to whatever you're exposed to in your environment. If you're identical twins raised in the same place, you're going to be exposed to the same things. But if I take identical twins and they're raised in different places, their immune system might have responded differently. And your immune system literally splices and resplices and adds in nucleotides and makes whole new genetic sequences for your lymph nodes. That's how you can have, for your B and T cells anyways, your lymphocytes. That's how you can have ability. You have a different B cell for any antigen you could possibly see. We're talking 8.5 billion different antigens, and you've only got 20,000 genes in your body. But it'll recombine your genetic sequence to give variability to each new cell. So those cells have a different genetic sequence than your rest of your body. Just really cool and wild, but this happens in all animals. And so technically identical twins aren't identical if they've been exposed to different things during childhood, or they had different gene reassortment or whatever like that. So we could be able to tell that technically if we were just looking at their lymphocytes, but you're not usually. You're looking at like a cheek swab or something like that, right? So anyways, if you're not genetically identical, then you will have a different pattern when I cut your DNA with restriction enzymes. So I don't even need to sequence your whole genome. I can just take your DNA, cut it with my restriction enzyme, and say, this is the killer, you know? So that's how they use that in forensics. Pretty easy tool there. But gel electrophoresis is good for that. It's also good for if you wanted to isolate a specific gene that you cut with that enzyme and you know what size it is, you could say, this is my gene, and then cut it literally physically out of the gel and purified it out of the gel. And now you have just that gene and not the other extra DNA. And so we use it for that as well. So that's gel electrophoresis. And then we can put those genes into cloning vectors. The main cloning vectors that we talk about in the chapter are plasmids, viruses or bacteriophages. Remember, bacteriophages are viruses for bacteria, or even artificial chromosomes. Those are the 3 vectors we can use to manipulate people's DNA. And even eukaryotic cells can have plasmids. They're just more commonly talked about in bacteria because we use them all the time in bacteria. It's how we get our bacteria to make insulin and things like that. And so we'll talk about that more in our transformation lab when we get to that one. It's lab 10. Okay. I don't want to get too trapped up in this. All right, single nucleotide polymorphisms. This is the last thing that we're going to talk about, I believe, on this chapter. Single nucleotide polymorphisms. Again, big words, fancy stuff, but it doesn't mean a whole lot. A single nucleotide is just one nucleotide. A polymorphism just means there's some changes. So that could mean that for the gene that codes for insulin, some people have an A here, but other people have a T. Some people have a G there, but other people have a C. Those are single nucleotide polymorphisms. It's just a change in one nucleotide within a gene, the different variations that you can have based off of just one nucleotide change. Sometimes one nucleotide change, like we learned with our previous chapter with our mutations, sometimes one nucleotide change doesn't change much. But other times, one nucleotide change can make it cause like a stop mutation or put in a different amino acid. Like if you're changing from a non-polar amino acid to a polar one or vice versa, that can drastically change the shape of your protein and how it folds. And then that can change its function and ability to work within the cell. Or if you have a stop codon, that can be literally deadly enough that like the baby wouldn't even grow. You wouldn't even know that you had a baby because the cells wouldn't be able to multiply, that sort of stuff. So like these single nucleotide polymorphisms can be devastating to the point that you wouldn't ever even have a viable fetus growing if they happen. But they can also be, you can live with them, but just have more risk. So it talks about in the book, like a thrombosis where you get clots because you have a single nucleotide polymorphism that leads to increased clots, leading to increased strokes and stuff like that. But one nucleotide change from the wild type, just one nucleotide, and it causes that protein associated with clotting to not work properly. So yeah. Okay. Chapter 9, I feel like chapter 9 is a lot of general stuff. With chapter 9, we had the trophs, and we basically have to organize these by carbon source or by energy. So there'll be questions on the test that will be asking about like, if this organism uses their carbon this way, or if the organism uses the energy, gets their energy this way, but we need to know the difference between the two. So if you get your carbon from a place or if you get your energy from a place. So we'll start with the energy one. If you get your energy from sunlight, we call that a phototroph. Easy. If you get your energy by breaking down, you're fine. Yes, thank you. If you get your energy from breaking down chemicals and releasing the energy in the bonds, then that is called a chemotroph. That's like us. So we break down our food and we get our energy from breaking those bonds. However, if you get your carbon from inorganic sources, like carbon dioxide, it's inorganic. Hate to break it to you guys, but it is. It's inorganic. There's no hydrogen backbone with it. If you get your carbon from an inorganic carbon source and you fix it into organic molecules yourself, we call you an autotroph. If you use previously made organic molecules like we do, we call those heterotrophs. So then if I tell you that an organism is a chemoheterotroph, this would be an organism that gets its carbon. When we're heterotroph, that's going to be it's from organic molecules that were previously made, and it gets its energy from breaking chemical bonds. This is us, right? We're chemoheterotrophs. We get our carbon from other organic molecules that other animals made by either plants or animals or whatever we eat. It already has organic molecules there. And we get our energy by breaking down our bonds in our food. Whereas plants are photoautotrophs. They get their carbon from an inorganic source, which is CO2, and they get their energy from sunlight. Photo. So don't overthink those terms. All right, moving on. We've talked previously about growth factors, so I'm not going to go over them again. Oh, come on, please. Why? But just briefly, I'll reiterate that a growth factor is something that you have to add into the diet of an organism because they can't make it for it themselves. And so the diet, whatever media that it's growing on or whatever food that it eats or whatever, we would call it enriched if it has that growth factor. And any organism that needs a growth factor, we would call them fastidious. So just reiterating that stuff, we learned about that previously, but just reminding. Okay. Saprobes, again, we've also learned about them, break down dead, decaying things. All right, then we're getting into our extremophiles. Halophiles, halo is talking about halogens. So halogens would be like chlorine, fluorine, iodine, things like that. And that'll be more important in unit 3. But a halogen would be fluorine, chlorine, whatever. But that includes chlorine, including sodium chloride. So halophiles flourish in salty habitats. Thermophiles, think of temperature. These are guys that flourish at high temperatures. Then we talked about diffusion and osmosis, and I feel like that was a big part of chapter 9. So we'll go over that really quick. Diffusion is anytime any molecule is going from a high concentration to a low concentration. So you have a pile of bouncy balls and they're in a net and you open up the net and they go everywhere. That's just diffusion, right? But if it's diffusion of water specifically, diffusion of water is called osmosis. Our membranes on our cells are what we call semi-permeable, and they are selectively permeable as well. So they're semi-permeable because they are selectively permeable, but they are selectively permeable to allow water and other certain small molecules, very specific ones, to just pass freely across the membrane. So whenever you put a cell into a solution that's high in salt or a solution that is lower in salt, only the water can cross. The other things that are in the cell can't, and things can't just freely move into or out of the cell, but the water can move across. And so that's why we talk about the movement of water specifically. And a solution is just going to be anything that is in a liquid. We have something dissolved into a liquid. The solute is the thing that's dissolved in the solvent, which is the liquid. And in biology, our major solvent, the solvent of life, is going to be water, right? And solute is whatever's dissolved in it. Okay, so we've got that. Selective permeability, we've talked about that. And so this leads into our concepts of isotonic, hypertonic, and hypotonic. So we'll draw our little diagram here for each. In an isotonic situation, if we put, this is our solution, and this is a cell that's inside of the solution. The concentration of what's inside the cell and the concentration of what's outside are equal. But the water is still always moving. It's just equal on both, equal in and out. There's not no movement, it's just equal. Equal in and out. In a hypotonic situation, hypo is lower. We know this, hypo and hyper, most people can get that part, right? But with the hypo situation, that means if a cell is put in a hypotonic solution, then the solute is lower. So the cell has more than the outside. So in this case, let's say it's pure water, which is the case with our cells, right? So our cells are not pure water. They might be 95% water, but if we put our cells in pure water, what happens is our cell says, oh, I have more bouncy balls in here than what's outside. I can't move the bouncy balls, but I can water down my bouncy balls, and maybe the bouncy balls will be equal concentrations then. So what happens is water goes in to try to water down what's inside and equal out the concentrations. In a hypertonic, we know it's higher on the outside. And so what happens in that is that you'll have your cell, which might have a low concentration of salt, but what's outside of it is super, super high. It still wants to help equal out and balance on that side, but only the water can move. The salt can't move in. So what happens whenever you put something in salt, and we know this because if you pack something in salt, what happens to the cells? The cells shrink. They dehydrate. Why? Because the water is moving out of the cells then. And so that'll cause the cell to shrink. This is a process that we call plasmolysis, and is the whole basis on us storing foods in high salt and high sugar. So even jams and jellies, they preserve well because the high sugar, and does the same effect as the salt does. So that's our isotonic and all of that. Our cells can't just only transport water, though. They have to be able to transport other things in and out of the cell. It's how we get our nutrients and everything into our cell, oxygen, and whatever else we need. So this is done by a process of diffusion that can be either literally diffusion, letting it happen on its own, but we make a special channel that things can diffuse through. And then if that's happening according to the laws of diffusion from a high area to a low area, then that would be facilitated diffusion. It doesn't need energy. Diffusion will drive it, but we'll facilitate it by creating a channel. It can't cross the membrane on its own. We have to help it out. We'll create a channel for it. That's facilitated diffusion. In active transport, we're moving against the gradient, or we need to move at a faster speed than what the gradient is moving it at. So if there's a limited amount of nutrients outside of your cell, and you've already store packed that nutrient, so you have more in the cell than outside, but you need more in there, so you're going against the gradient, then you would need active transport, and that's always going to require energy. All right. Organisms that are able to live at certain temperatures. So our temperature tolerance, we can actually define our organisms by the temperatures that they thrive at. Psychrophiles, I don't know where the word comes from. I don't know the origin, so don't ask, but psychrophiles like it cold. Mesophiles like it medium. Meso, medium, neutral. This is like room temperature, and all of our pathogens, they live at the same temperatures that we do, middle temperatures. So those are mesophiles. And then organisms that like it really hot beyond that, and we're talking like 100 degrees, like well beyond, like even past boiling. These will be our thermophiles. We talked even today in lab, we talked about the toxic oxygen products and hydrogen peroxide being metabolized by catalase. And then superoxide dismutase is an enzyme that deals with superoxide. So these are our toxic oxygen compounds. We kind of touched on that today in lab, so I'm not going to go over it much now. But we can actually define our organisms based on their ability to survive or live with or even use oxygen. And so this would be our aerobes, aero, air, they live in the oxygen. Facultative anaerobes. Don't read too much into the fact that it says anaerobe, because these guys are technically aerobes. It's just that whenever there's no air, they can go through fermentation. And so I focus on the F on this one because facultative anaerobes ferment. And so if there's no oxygen, they'll ferment to make their energy, which of course we're going to get to in chapter 10 in a little bit. So those are our fermenters. Microaerophiles, they don't need as much. They need a micro air, small amount of oxygen. Aerotolerant anaerobes. These guys never use the oxygen, but they have defense mechanisms against toxic oxygen products. So they can tolerate the air. And then obligate anaerobes, these guys cannot do the air. They will not do the air. They don't use it. They're not equipped for it. These are like C. diff and stuff like that. All right, the last little bit of it kind of talked about symbiosis. Symbiosis is any time that two organisms' livelihoods are intertwined somehow. If an organism depends on another organism for survival, that's a symbiosis. If we have an impact on other organisms in our lives, but we don't depend on it, that's not symbiosis. Okay, but yeah, symbiosis is dependent. So there's three major types of symbiosis. Commensalism. Commensalism is where one benefits and the other is unaffected. The second one is mutualism, and I feel like it kind of speaks for itself. Mutual benefit. Both are benefiting. And then the third one is parasitism, parasite. One organism benefits at the expense of the other. One is helped and one is harmed. Always if one of them's being harmed, that's parasitism. And then that's where we get to that idea of obligate parasites, like viruses. Viruses, one of the first things we introduced with them. They were obligate parasites. They're obligate intracellular parasites. They're parasites because they are using our resources and our cell parts and components and everything to build. And so it's at our cost. It's harming us. But they're obligate because they can't do any of that without the host. And this also includes things like ticks. Ticks can't live without feeding off of the blood, right? And so they are also obligate parasites. But a lot of our pathogens that are true pathogens kind of fall into that category. All right, next we have our biofilms. We learned a little bit about how biofilms are formed as a result of our capsules and slime layers. But some organisms can actually like read like the waste products or signaling molecules that other ones are making within the biofilm and kind of communicate with each other. And that's quorum sensing. Ability to communicate within that biofilm. The last thing that the chapter talked about was our bacterial growth curve. We do need to know the different phases of bacterial growth curve. There absolutely is a question about that on the exam. So the 4 phases are, and it looks like this. So up, flat, down. The first part of it is the lag phase. The lag phase is just where the bacteria are just getting accustomed to their environment. They haven't started multiplying yet. They're just sensing their environment and getting accustomed to it and adapting to, being in a new place. Once they start actually doubling and doubling and doubling, this would be the logarithmic, literally, or exponential phase, where we're growing and doubling and everything like that. Then we have the stationary phase. Stationary phase is where growing equals death, or where we're not growing as quickly. We're running out of resources, and some guys are just dying off of old age. And some guys are reproducing, but we flattened out. That's the stationary phase. But the death phase is literally like what it sounds like. You have more death than you have growth by far. And eventually everybody will die when we run out of nutrients or if there's toxic products from just death. All right. That's it for chapter 9. Come on, dude. Chapter 25, you can see how I feel about chapter 25. Okay. It's pretty easy. There's the carbon cycle and the nitrogen cycle. We have the 3 main types. So I'm skipping over the carbon cycle. I feel like that's fair, right? Can we all agree that the carbon cycle is pretty straightforward? Carbon cycle, carbon dioxide is used by our autotrophs to basically fix it into organic molecules. All the other heterotrophs use that and exhale carbon dioxide, and it just cycles like that. Of course, we have environmental stuff that feeds into that as well, but it's mostly carbon dioxide or organic molecules and just switching between those. So easy enough. Methane is included in that. And some of our bacteria produce methane as a byproduct. It's actually a worse greenhouse gas than carbon dioxide. Cool, we said it. All right, nitrogen. People have issues with the ammonification, nitrification, and denitrification stuff. I am not going to show you equations. I am not going to ask you about those things. Just be aware. Here's all you need to know about nitrification, because I know this is a tricky thing for people. Just be aware that these 3 are ways of that organisms can deal with nitrogen in our environment. I'm not going to ask you about the difference between them. Just be aware that these 3 things are part of the nitrogen cycle and are necessary for nitrogen to be cycled in our environment. It's actually pretty important in soil biology. And it's a huge problem right now, especially with global warming. I don't care how people feel about global warming. It is actually happening, regardless of why or how. I don't care, but it is happening. And it's getting so hot in some parts of the world that the bacteria can't live because of the heat, and the bacteria that deal with these 3 things and make nitrogen usable for plants aren't able to live. And so the plants can't grow without the bacteria. That's a huge problem right now. In some areas of the world are starting to starve because they can't grow their food anymore because bacteria can't grow because it's too hot. So it's an actual real problem in the world right now. But these cycles, ammonification, nitrification, and denitrification, I won't ask you about the specifics about each of them, but know that they're important in the cycling of nitrogen in the environment. That'll be enough.
Question on the test would be,
which of the following are cycles that, yes, are important for nitrogen? Exactly. And I won't ask you about each of them individually or what they are defined as. We've mentioned them. We're aware of them. That's the most I need. So I can't be bothered to care beyond that. Okay, so our marine microbes and plastic. Plastic creates reefs in the ocean where microbes can create little microbiomes. And I say little, but we're talking about plastic that builds up in the ocean. It's literally the size of the state of Texas. There's an island of plastic that is somewhere near like India in the Pacific Ocean that is literally the size of Texas. Like that is gross, but it affects everything underneath it, so weird microbes are growing there. And then the organisms that would feed off of the normal microbes are getting, either poisoned or the other ones can't grow or their food sources are disrupted. And so whatever. So we're talking about massive ecosystem disruption just because of plastics floating. And so those are the macroplastics, but there's also microplastics and the ramifications that they have and how they're affecting our environment as well. So it's fine. It's not fine, but you know what I mean? Like it's a problem. All right. So water filtration, we have two processes that we talked about in this chapter. Water filtration for the water that we drink and use on a daily basis. Clean water. And then we have wastewater treatment. Wastewater treatment is what we do to deal with sewage. Right. I'm not going to go through the steps of those different things. And you guys don't need to be super specific about remembering all of the steps and which ones come where and whatever. But definitely know the difference between these two different types of filtration and why we have more steps in the wastewater, right? So the whole point of the wastewater filtration is to release the water back to the environment so that it can not be containing poop in it anymore, right? So we have to remove the poop. And so there's more steps in treating that because the clean water that we're getting for our drinking usually comes from a relatively clean water source. Even if it's lakes or rivers, it's not contaminated with poop that we know makes people sick. And so the bigger issue with testing these and making sure that they are safe for people is looking for the presence of coliforms. We call them fecal coliforms. These are bacteria that we know are present in poop that might not cause you disease in small numbers. They might not cause a healthy person disease in small numbers, but if they made it through our filtration process, the pathogens can too. So things that are bad E. coli, because we talked about there's like a million different kinds of E. coli, it's ridiculous, but there's a whole bunch of different E. coli's. The really bad E. coli, the S-Tech E0157H7 that we hear about on the TV and all that, or Shigella or cholera or Giardia, those things can make it through if Klebsiella made it through, if Enterococcus made it through. You know what I mean? So like normal poop bacteria make it through, the bad guys probably could too. So we test for the normal guys and we have allowable limits of each of them. And we call it basically colony forming units and how many colony forming units are allowed in drinking water versus like watering a park or releasing it back in as wastewater that's been treated. So the ones that are released as wastewater that's been treated will have the highest number of coliforms, whereas drinking water should have none, for example.
So what kind of water goes into like splash pads?
Usually somewhere in the middle.
Okay.
Yes. Yeah, it's somewhere in the middle. It's not as usually as clean as drinking water is, but it's cleaner than by far than like our treated sewage. Yeah. Right. Using microbes for food and stuff. I don't need to go over that. And then at the end of it, it talked about One Health, which is the idea that the health of the world affects the health of the world. Like, how else can I put it? It's sort of like if the animal populations are suffering, I think even better, like one that we talked about earlier, if the mosquitoes are gone, then the things that eat the mosquitoes won't be able to survive. And the things that eat that won't be able to survive, and so on and so forth. So looking at the health of like a population versus the health of a part. Okay. These are, of course, like the longest chapters, but they all come together. All right. Chapter 10, anabolism and catabolism. Know the difference between the two. Cats destroy. I say it in my lectures all the time, but that is how I remember. Cats tear things apart. Catabolism is breaking things apart. Anabolism, like anabolic steroids, is building. And so when you tear something apart, you release the energy from the chemical bonds. So catabolism creates energy. Cat creates energy. I don't want people to confuse. Cat destroys things to create energy. Okay. Anabolism is building things. And anytime you're building things, if you're building a whole building, for example, you would need energy to build that. So anabolism uses the energy. And that's where amphibolism comes into play, is this the tying together of breaking things down to get the energy to build other things. And so it's how those two things tie together. And then metabolism is even bigger term. Metabolism is just all of the chemical reactions that happen within the cell or even in a body that's multicellular. So enzymes are proteins that are usually coded for in our genes. They're products of our genes that will help act as catalysts in chemical reactions. And a catalyst is basically anything that lowers the amount of energy to allow a chemical reaction to occur. and just period. That's what a catalyst is. And so these are proteins that act as catalysts in our cells, and they are not changed permanently or anything. They can be reused over and over and over again. No permanent changes are done to them, but they will work to make permanent changes to whatever they're acting on. The thing they act on is called a substrate. So enzymes, they are catalysts that act on substrates, usually cause the substrate to be chemically changed. So you could take two substrates and make them into one product, or take one substrate and turn it into two products, or a lot of other things. We can modify it or whatever we need to do. Enzymes can be either simple or they can be like conjugated, which are more complex. If they are conjugated, then they exist of two parts. The two parts will be the apo enzyme, which is the protein portion, or the second part is the cofactor. This is the non-protein portion. And the non-protein portion can be inorganic, or that can be organic. If it's organic in nature, we call it a coenzyme. So a coenzyme is a cofactor. It's just an organic cofactor. When we have metal ions that act as cofactors, like we do with the iron in our blood, so like hemoglobin is a protein that binds with iron in order for it to function. So iron is a cofactor, but it's not a coenzyme, right? It's not organic, but it is a cofactor. It's just not a coenzyme. And so a lot of the molecules that we talk about later in this chapter, NAD, FAD, whatever those are organic molecules that are acting to help with the enzyme process later on. So those NAD and FAD molecules, those are coenzymes. So coenzymes are just organic cofactors. All right. Once you have combined your apo enzyme and your cofactor together, that creates a molecule that we call the holoenzyme. This is the whole enzyme. It's just with an H there, but it's holoenzyme. That's the whole active functional enzyme. The area of the enzyme that acts on the substrate is always going to be in the protein portion, the apo enzyme. Always. So the part where we're actually interacting with the substrate, that takes place in the apo enzyme at a site that we call the active site. All right. Clear it up. We'll clear all of it, sure. Okay. Moving on to our enzyme classes. There are really only 4 that I'm going to test you on. There's 6 of them to know that exist, but I'm only going to test you on 4 of them. And I feel like the first one's the obvious one on this list, but like we'll go over the other ones first that I care about. Transferases speak for themselves. They're just transferring a group from one molecule to another. They're literally just transferring it, moving it from one place to another. Hydrolases and ligases are kind of opposites. We're going to talk more about hydrolysis whenever we get into lab 13. We're going to do, I don't know if it's 13, 14, something. It's one of the labs, 14, I think. Lab 14, where we're going to look at hydrolytic enzymes and whether our bacteria have the ability to hydrolyze starch or hydrolyze urea or hydrolyze gelatin. And that just means breaking those things down. And so hydrolysis is a breaking down process. Hydrolase is an enzyme that helps with it. So this is break down. And usually what's going on is you might be a molecule that's AB that we're going to be breaking down to A plus B. But we're using water to do that. So we'll have H2O and we'll break it apart. And then the A will have an OH and the B will have an H. And so we've lysed water. So that's what hydrolysis really means. We're breaking water to break apart other things. So that's what hydrolysis is. That's what hydrolases do. They help with this process. Then the ligase, we already learned about DNA ligase, right? We know ligase. What does it mean? It means to bring it together. And so in order to go from A plus B and turn it into AB, guess what's going to happen? The A that had the OH and the B that had the H, to become AB, they're going to give off water. It's literally the opposite. Okay. So that's why I always try to bring them together, because they kind of happen at the same time. So in order to build molecules, we'll create water as a byproduct. In order to break down molecules, we'll break down water to help break apart the other products. So that's how ligases and hydrolases are related. And then the other one was oxidoreductases. I mean, yes, if I talk about an isomerase, whatever, it's changing just the orientation of a molecule, I'm not going to ask you about it. I'm not going to ask you about liases. That has to do with double bonds. But I am going to ask you about oxidoreductases, because obvi, that's like pretty much what the whole chapter relies on, is oxidation and reduction. We've kind of already talked about denaturation, so I'm not going to talk about that again. Denature just means to break down the structure of something, especially with proteins, that often relies on disrupting hydrogen bonds. Hydrogen bonds can be disrupted by heat, like we learned about with DNA. It can be disrupted by pH as well. Too many H pluses around, you have a problem. Too few H pluses around, you have a problem with your hydrogen bonding. So pH, whatever, pH can have an effect on that, on hydrogen bonding, and lead to denaturation as well. I mean, so that's one way we can control our enzyme, is through denaturation. I'll mark these off as I go so I don't get lost. All right. In order to inhibit the function of our enzymes, we can do that either at the genetic level, which we kind of talked about with operons and bacteria. They have similar functions that happen in eukaryotic cells. They're just different. They're not operons. But in the case of the actual enzyme itself and inhibiting its action, you can either interact directly with the active site. So if you are an enzyme and you look like Pac-Man and you act on triangle pizza, but I bring in a drug that looks like triangle pizza, and the drug comes in and binds here instead of triangle pizza, now the enzyme can't interact with triangle pizza. And so this is what we call competitive inhibition. It's competing for the active site with the substrate. For non-competitive inhibition, if you were that same enzyme and you normally interact with triangle pizza, and then something else comes along, let's just say it's a square, whatever. Here's our square. Square comes along and binds somewhere else on that, and that leads to this situation. It causes the shape of that enzyme to change, but it didn't bind at the active site. It bound somewhere else, right? We'll say it was right here. It looks like a butthole, but you get it. So it bound here, and that site is the site that we call the allosteric or regulatory, but same interchangeable term. And this is non-competitive inhibition. Results the same. We can't bind with triangle pizza anymore, right? Triangle pizza can't fit in here anymore. But we just didn't bind at the active site. So that is competitive versus non-competitive inhibition. Are we ready? Because I think we know what comes next, right? So this is going to be, good God. This is going to be talking about redox reactions. And then we're going to get into the actual metabolism. And I'm going to open up a whole other window for the metabolism stuff, because I want to give it its due diligence. Because this is the part, right? Like this is hard for people. Okay, so first is redox reactions. I know we learned about them in chemistry, but I want to simplify it for you guys. The way I remember it, I don't care how you guys remember it. It's either oil, Rig, or Leo says Gur. I learned Leo says Gur. And literally, when I do chemistry and somebody's asking me about reduction or which one is oxidized or which one's the oxidizer or whatever, I have to write down Leo Gur, because I'll be like, okay, I have it written down. Now I can think of which one is the oxidizing and whatever. So starting with oil rig, oxidized is loss. Oxidized is loss. And then reduction is gain. Or Leo says GER, which is lose electron is oxidized. Or gain electron is reduced. We got it. That's how I remember it. And there's definitely gonna be questions about that on the test. So we've covered it. All right. ATP, aerobic respiration, and all of this. Cool. So ATP is our energy molecule, and it looks like, I'm not going to draw the whole thing, but you have your nitrogenous base, which is your A, attached to a sugar, which is a 5-carbon sugar that we know fondly and lovingly as ribose. And it is attached to a phosphate and another phosphate and another phosphate. It's triphosphate. And these phosphates, man, and they're not just a P, they're a PO4, PO4, PO4. And these oxygens, man, they hog electrons and they're so freaking negatively charged all over the place. They've got all this negative, 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 negative. And the negatives don't like being near the negatives. And even though each phosphate is covalently bonded to the other phosphate and they are sharing electrons, they don't want to have anything to do with one another. And so those phosphates, even though covalently bonded, are constantly battling and pushing against one another. So when you clip one off, it releases all this energy and it allows it to drive the production of the cells. And so how that actually works at the chemical level is you're just moving that electronegativity around. So this guy is super negative against the other negative guy, and we'll move part of that negative onto another molecule. That negative being on that molecule causes that molecule to change, and that drives the actual chemical reaction that's going to happen downstream of that. And so it's literally just moving the negative electronegativity of phosphate around that causes molecules to change chemically just by being near that negative. And that's what actually gives the energy that drives everything. So it's just electronegative charge, more or less, but moving that around. And so to create ATP, which is our major source of energy for the cell, we have a lot of processes in the cell that kind of work together to do that. And this is where I'm going to switch to another blank thing. It's blank. I've got it kind of already listed out, but we'll start with glycolysis. And so these are the 3 major steps in respiration. I don't care if it is aerobic or anaerobic. These are our 3 major steps. There's only one difference. We're going to get to it. Glycolysis is the first step of cellular respiration. Glycolysis is just the process of going from a 6-carbon sugar, which is usually, like it implies in glycolysis, glyco, what do we say about it? We said it's sugar. 6-carbon sugar, it's usually glucose. And we're going to be breaking down glucose into, we have 1 6-carbon sugar that we will break into 2 3-carbon pyruvate or pyruvic acid, depending on how you're looking at it in biochemistry. I will not make any distinction between pyruvate or pyruvic acid. Same thing in my book. Okay. So we are breaking down a 6-carbon glucose into 2 3-carbon pyruvates. If you've looked at the book or if you've learned about aerobic respiration, you know it's not that simple. There's like 50,000 steps to get to that pyruvate. And we're giving off stuff in the middle of that. Some stuff that we'll be giving off, we'll give off some NADH. I don't care how much, I'm not going to ask you. And we will give off a net of 2 ATP. So these are some products that we'll make in glycolysis. We'll make a little bit of NADH and ATP, just a little bit. Glycolysis is where fermentation stays. So in fermentation, this is the only source of ATP. This is a big deal to remember, especially when we compare how much freaking ATP we're going to get from the electron transport chain. When you ferment, if you have oxygen, you can go through steps 1, 2, and 3. But if you are a facultative anaerobe and you do not have oxygen, you ferment. And that means you're only going to make 2 ATP per glucose molecule, as opposed to the 38 that you could make if you went through the whole thing. So fermentation just stays here. That's the only step that fermentation does. The next step, if you're going on from here, you have to convert your each of those. And I'm just going to put each in here so we will remember, but each 3 carbon pyruvate is going to get converted into 1 acetyl-CoA, which is 2 carbons. 2 carbon acetyl coenzyme A, which we also call just acetyl-CoA. But it's 2 carbons. What the heck? Where did the other carbon go? Easy. Let me help you out. We're going to make some ATP here or some NADH or something like that. Who cares? But the other thing that you're going to make is carbon dioxide. This is our first carbon dioxide molecule. And I would like to kindly point out to everybody that's watching the video that when you exhale, like I just breathed in, I breathed in oxygen out of my exhale. That carbon dioxide didn't come from the oxygen I breathed in. It came from my glucose into the pyruvate. That's where the carbon dioxide that you exhale comes from, period. That's the place. All right, so next step is the Krebs cycle. The Krebs cycle is going to take acetyl-CoA, which is why we need to mention the intermediate step. Otherwise, where did acetyl-CoA come from? But we're going to take our 2 carbon acetyl-CoA and we're going to cycle it through some stuff. This is also known as the citric acid cycle because one of those molecules that we're going to be making is citric acid. So in the Krebs cycle, we're combining our 2 carbon BS with some 4 carbon BS to make some more 6 carbon BS. It's like citric acid. It doesn't really matter. I'm not going to test you on each of the steps. I'm not. I promise, and you're welcome. Yeah. I don't think it's important. What I think is more important that we understand is kind of why this is happening. So this is happening. We're going to put our 2 carbon with our 4 carbon and make a 6 carbon. But how do we get the 4 carbon in the end? Because we're kicking off what again? Carbon dioxide again. So we'll do this a couple times. CO2. And so that's how we get from our 2 carbon plus our 4 carbon. What is our waste product from the last cycle? Make our 6 carbon, kick off some carbon dioxide. Great. Is this the whole point of Krebs, though, is to make carbon dioxide? What are we getting out of it? That's not the point of it. The point. So we already said the point of glycolysis. The point of glycolysis is to make pyruvate. The point of the intermediate step is to take that pyruvate and make coenzyme A. The point of Krebs is to take that coenzyme A and make our other byproducts, NADH and FADH2. And so what are these guys really? These are just electron carriers. Whatever. These are literally just there to carry electrons around in the form of the H. The H's. So think of the H's as being electrons. Because once we start dealing with them in the electron transport chain, we're going to start seeing them as H pluses. But are H pluses just H pluses? They're H pluses, sure, but they had to have had. Why does it do that? H pluses had to have had an electron at some point. They weren't always just H pluses, right? They used to be linked to NAD. They used to be linked to H2O. They had electrons with them that they were sharing with other molecules. So these guys are literally just sharing those electrons with the NAD or the FAD, but the electrons are there, okay? So these are our electron carriers. This is the point of the Krebs cycle. This is the point of the Krebs cycle. So pyruvate we need to get from glycolysis. We made a little bit of ATP. Yay. Like, I don't know what to tell you. Like, yay, a little bit. We made a little bit here too. We made a little bit in the Krebs cycle, ATP. A little bit, like 2. Yay. But we want to make more than that. We need more. That's what the electron transport chain is going to do for us. So we are going to take electrons that we saved up in these spots. We have some here. We have a little bit up here, our electrons. We have a lot from the Krebs. This is where we got most of our electrons from, is from the Krebs. We make carbon dioxide as a byproduct of that. It's not even the point of it. We just wanted the electrons. So we're gonna take our electrons and turn them into ATP by creating a proton gradient. I'm gonna draw it out, so stick with me. This is how it works. Here is our membrane. And we know how the membrane really looks like, right? You've got your phospholipids. So whatever, this is our phospholipid bilayer. Just imagine that it's there, okay? So here's our membrane. And then we have these things called cytochromes on the membrane. I'm gonna make them green here. It doesn't matter what color they are. I'll put 4. And these green blobs that I'm putting here, these are called cytochromes. And we'll have this other thing at the end of all of this. Looks like a channel. Draw a little bit Minecrafty, but that's okay. This whole thing is a unit, like it's like a channel that things will go through, but this is like one big old unit here. This orange thing is called ATP synthase. It tells you what it does. It's going to synthesize ATP. And so what happens is we will get our NAD or our FADH, our NADH and our FADH2 that we made from the Krebs and the glycolysis. Whatever. So we've got all of these guys, and these are our electron carriers, just to remind us. And so how are they carrying the electrons? So what these guys are going to do is make the electrons come off. Oh my gosh, please just work with me here. Electrons will come off, and we will also have something else come with it, which are our H pluses. So now we're looking at them as separate entities. If they were together, it would just be H. But now we're separating the electrons and the H pluses. All right. And then our byproducts of that will be just our NAD and FAD. And they'll go back into that other crap. So those are our coenzymes that are electron carriers. But what we really need are our electrons and our hydrogens. So what happens is the electrons will excite these cytochromes. And when the electrons having a negative charge interact with the cytochromes, this causes the positively charged hydrogens to move out, and they'll collect on the outside of the membrane. I'm going to put a bunch of pluses out here so you know that they're out here. And I mean, they'll create this massive gradient of plus on the outside. They're really H pluses, so I'm going to draw that here. And this is called the proton motive force. And this process by which the electrons are causing this to happen, this is called chemiosmosis. So the electron goes through one cytochrome, it causes a proton to be pumped out. It moves on to the next cytochrome, and then the next one, and the next one, and so on. Each time, hydrogens are getting pumped out, creating this gradient, the proton motive force. We following? I hope, kind of, okay. Those negatively charged electrons causing the cytochromes to be proteins. What happens whenever we have all these amino acids, these different amino acids on our proteins coming into contact with a negatively charged thing? They'll change their shape. That allows the protons to move out. And so they get pumped out that way, and we create this positive gradient on the outside. Well, if it's way more positive outside than it is inside, what did we learn about diffusion? Well, it wants to equal out, right? And so because all those positives that are outside want to come back inside to equal things out, they do. They come inside. As those positively charged things move through this whole other protein, again, proteins are made of amino acids. They've got glycine, they've got leucine, they've got phenylalanine and whatever. And some of those charged guys, when they interact with that positive passing through it, this allows for ADP to get phosphorylated with an inorganic phosphate. Say inorganic, it's a PO4 that's just not bound to something, but to become ATP, which is the goal. This is the goal. This process alone will make 32 of these. From one glucose molecule, you'll get 32 ATP here, which is why when we talked about fermentation previously up here, 2 ATP isn't great, but it'll keep them alive without oxygen. But why do we need oxygen down here? Okay, so I'm so glad you asked that question. So all these electrons that move through the cytochromes, they're building up now. All these hydrogen, these positive hydrogens, they're building up here. So what's going to happen is you breathe in, you bring it in on your red blood cells. They're being carried by your hemes on your red blood cells. We bring in that oxygen. We take an H plus, 2 H pluses, and 2 electrons. Those give us just H's, 2 H's. And then we will take a half of a O2, which is 1 O. So that's where we get our H2O. So when you inhale oxygen, the only thing it's there for is to get rid of those electrons that moved through the cytochrome. We're done with them. And to get rid of those H pluses so that we don't give a buildup of H pluses on the inside of our mitochondrial membranes, in our mitochondrial matrix. So in eukaryotes, this is all happening. On the mitochondria cristae. So I just put mitochondria. I'll put cristae. And the inside is the matrix. So what I'm saying is that if this is your mitochondria and this is your cristae on the inside of your mitochondria, and this is your matrix on the inside. And so your H pluses are all in this space here. This is what your mitochondria really do. You say it's a powerhouse and it makes ATP. This is what you really mean when you say that. It's doing this. That's what your mitochondria are really doing. And so the whole reason, the only reason that you breathe in oxygen is so that you can get rid of those electrons and those hydrogens. These. It's just going to delete them no matter what I do. That's the only reason you breathe in oxygen. And when I suffocate you, you die because you can't keep doing this. Okay? That's why I stress this so much. Is like, we're learning about this in the context of bacteria, sure. But this applies to all living things that need oxygen to breathe, including us. And so this is why maintaining blood pH is important. It's why making sure that your patients are oxygenated because of ATP, and then maintaining your protein stability and stuff too. So anyways, so that's our eukaryotes. This happens in your mitochondria. But in prokaryotes or eukaryotes, they already look like this, right? So they already have an inner membrane. This is just happening on their cytoplasmic membrane or inner membrane. And their ions just build up on outside of their membrane. It looks just like our mitochondria, though. They're single celled that already look like the mitochondria. Okay. Do you have any questions? Okay, I know it's one of those ones that you kind of have to work through and make sense of it for yourself. So I'm going to move on, but this is my explanation for it. And I hope that at least helps somebody that ends up watching this. So I'm going to go back to my slides. Again, we already talked about amphibolism. We talked about the difference in these. We talked about the products. We already made it through all of this. Fermentation, again, if oxygen isn't available, they just stay in glycolysis. They still make NADH, but they can't put that in the electron transport chain. So in order to get rid of the electron being carried by NADH, they feed it back into pyruvate. And they convert pyruvate into either lactic acid, alcohol, or carbon dioxide. So that's why alcohol, lactic acid, and carbon dioxide are common products of fermentation, because they're just have nothing to do with the NADH that they're making. So they have an electron. And so they feed it into pyruvate, which was another product from glycolysis. And it just changes pyruvate into literally ethanol or lactic acid, just depending on how they deal with it as bacteria. So that's where those products come from fermentation. Alcohol and acid. All right, photosynthesis is also kind of wild. I guess I could do another thing for it, just really quickly. Photosynthesis has two parts. The light reactions and what we call the dark reactions, also known as the Calvin cycle. Light independent is really what the proper term would be. All right, so in the light reactions, this is triggered literally by sunlight. Sunlight comes down onto, let me go ahead and just draw this for you because I feel like this part is important to understand too. Does this look familiar? This should. This should look very similar to what we drew up there, right? Let me put my little channel in here. I'm not going to color it all in or anything, but you get the idea, right? Okay. Light comes down, hits these guys. These are not cytochromes this time. This time, these guys are pigment molecules, like chlorophyll. And when they get hit with the light, that excites the electrons that are already in the chlorophyll. And they do the same dang thing, wouldn't you know? Excites the electrons that are in the chlorophyll to move through the same freaking thing and cause the same thing to happen. H pluses to move out. And we get a bunch of pluses up here. It's the same. It's just instead of us bringing electrons there, we're exciting electrons that are already in the pigment molecules. Okay. And then these pluses come back in. And then what do we make as a product? ATP. This ATP, though, this is not enough to power a whole cell. And it doesn't give them their organic molecules that they need to build their cell, because they aren't getting their organic molecules from their environment like we are. They don't break down food to get organic molecules, so they still need organic molecules. So in order to make their organic molecules and create even more energy for themselves, they use this ATP. Let me redraw that. To drive going from CO2 to, y'all, glucose. What do you think they do with the glucose? I hate to break it to you. They do all this. They do this. They make glucose from light. They take light, make a little bit of ATP, use that little bit of ATP to go from a 1 carbon carbon dioxide to a 6 carbon glucose. And then they just feed that freaking glucose right back into glycolysis and Krebs cycle and the electron transport chain, where they're going to need oxygen in order to keep that side up. So I hate to break it to everybody. Plants don't just have chloroplasts. They have chloroplasts and mitochondria. And how do their mitochondria work? The exact same as yours. Can you suffocate a plant? Oh, you bet. You bet. Plants can't live in areas where there's no oxygen. So I think it's wild. I feel like people don't know this. And I feel like this is one of those things that's like also important to remember. We need way more than they do. They can make a little bit of their own oxygen from this process right here, because this process also gives off O2. And so they can use their own oxygen to feed that, but it will become self-limiting at some point, because it just doesn't make enough ATP for them. And like all the other processes in their cells, they're going to need to make more ATP. So this will eventually go through aerobic respiration. That's why I wanted to draw this out, because I just wanted to remind everybody, like, there's two parts to photosynthesis, the light cycles and the dark cycles. The light cycle is very similar to the electron transport chain. It's just instead of electrons, we're using sun to excite pigments. But then in the dark cycles, we'll use the energy from photosynthesis to drive fixing carbon dioxide into 6-carbon glucose, and then make even more ATP to power building cells and the other molecules in the cell. It's a lot, but yes, but that's how it works. Okay, moving on to the questions, and we're going to run through these. If you guys want more information on each of these topics, they are on the slides. So please, you can download the slides and look at them yourselves. But I'm just going to move on to the questions. Which of the following statements accurately differentiates bacterial transcription from eukaryotic transcription? Well, bacterial versus eukaryotic, bacteria have no nucleus. Already, I'm just going to write that down. And eukaryotes have a nucleus. We already know this is our basic difference. Let's see what we can find from that information looking at this. Bacterial transcription lacks RNA. We haven't talked why would have or RNA polymerases? No, if they have to make RNA from DNA, that general thing is still the same. So it's not this one. Eukaryotic transcription occurs in the cytoplasm. No, eukaryote transcription. Remember, transcription was DNA to RNA. And DNA is always, period. In a eukaryote, DNA is always in the nucleus, period. So it can't occur in the cytoplasm, which is outside of the nucleus. Anywhere outside of the nucleus is the cytoplasm. So this is not B. Eukaryotic transcription involves extensive mRNA processing. Okay, that's one thing we didn't list in our notes, but this is true. What kind of processing? The splicing of the exons and introns. So this is matching up, but let's rule out our last one. Bacterial transcription uses introns and exons. It's the opposite of what this is saying, right? So C is our answer here. A new drug is being tested for its ability to inhibit transcription in bacterial cells. Which of the following mechanisms is the drug most likely targeting to achieve selective inhibition? Something where transcription is different in bacteria versus eukaryotes. Is it targeting DNA polymerase? Is it targeting the ability to shuttle mRNA out of the bacterial nucleus? Bacteria don't have a nucleus. We'll come back to A. I just want to look at my other options. The RNA polymerase activity in bacteria, or the splicing of introns from eukaryotic mRNA transcripts. Well, if it's a thing that inhibits bacterial cells, it's probably not D, because they don't have introns and stuff. So it's not going to be that one. So now we have the difference between DNA polymerase in bacterial cells or the RNA polymerase in bacteria. So if we're inhibiting DNA polymerase in bacteria or the RNA polymerase, and we're worrying about transcription, we're not worrying about replication. DNA polymerase is for replication. And so now this brings us to a default of the RNA polymerase activity in bacteria. So this one doesn't have the direct answer. We had to find this answer by a process of elimination. And I don't typically have questions like that on my test, but if we could think through this here, we should be better prepared for that. As it turns out, the polymerases are slightly different for RNA or for bacteria versus eukaryotes, but we had to answer that through process of elimination. The next part is reminding us about our operons. So I'm just going to write up here again. If you are inducible, you are off. And you can be turned on. Can be induced, right? Repressible operons, these guys are on and can be turned off. All right, see if we can answer our questions. Which of the following statements accurately describes a key difference between the inducible and repressible operons? Inducible operons are typically off. Okay, yes, that matches, and are turned on by an inducer. We would call it an inducer because it's causing the repressor to fall off. So it does induce it. While repressible operons are typically on and are turned off by a repressor. Okay, I'll give this one. We'll come back to it and see if it's a problem or not. Are inducible operons always on? Well, according to my little mini note up here, no. So I'm already knocking that one out. All inducible and repressible operons are regulated by lactose. Common choice from people. Don't get mixed up, okay? The lac operon is an inducible operon. It's not repressible. Inducible operons and repressible operons both use the same type of repressor protein. Each operon, so there's an operon that controls for lactose breakdown. There's an operon that controls for making arginine. There's an operon for this, and there's an operon for that, and there's an operon for this. Every single operon has its own repressor, and they're individual. So that part is wrong also. So A is our correct answer. A pharmaceutical company is developing a drug that aims to inhibit the expression of genes involved in bacterial antibiotic resistance. The drug specifically targets a repressor protein. So I'm going to draw my little thing out real quick just to remind us where we're at. So in DNA, in an operon, you have a promoter region, an operator region, and you have your genes that are downstream. And then the repressor binds the operator to block RNA polymerase. So it can't go. That's how operons work. All right, so which of the following would be a likely outcome of this drug's action on the bacterial cell? So if it's regulating a repressible operon, one that is on and can be turned off. Will the operon remain off regardless of the presence of the repressor? Or will it be activated in the absence of the repressor? It's not going to remain off because it's already on. If you're messing up the repressor's ability to turn it off, I'm imagining it's probably not going to be able to turn off. That's the real problem. So it's not going to be A. B, will it be activated in the absence of the repressor, leading to increased expression of antibiotic resistance genes? Maybe. I'm leaning towards that one. C, the drug will have no effect? No. Or the operon will be turned off regardless? No, it's going to stay on. So that's why I'm going to say B, the operon will be activated. You can't turn it off because your repressor can't block it from and make it stop. And that's what's really going to happen. Probably not a great drug to use, if I'm being honest, based on that. Okay, I'm trying to go to the next. You know, sometimes it goes to the next screen so easy. There it goes. All right. Questions for chapter 8 stuff. The first one has to deal with restriction enzymes, the ones that cut at the palindromic sequences and create the sticky ends. Palindrome and sticky ends. Question 1. Which of the following best describes the role of restriction enzymes in molecular biology? Are they used to amplify? No, they're used to cut. B, they cut at DNA at specific sequences, creating fragments with sticky or blunt ends. They can do blunt ends too. We didn't talk about it, but they can. Do they synthesize new DNA strands? They don't synthesize, they just cut. Or do they bind to RNA and inhibit? No, they cut. So B is our answer. Next question, a clinical laboratory is using restriction enzymes to diagnose a genetic disorder. The disorder is caused by a specific mutation that alters a restriction enzyme recognition site. So instead of cutting at that site, now we have a lack of a cut. Lack of a cut is what it sounds like. How would this mutation affect the results of the fragments that result? So I'm imagining if you're not cutting at a place, you're going to have bigger fragments. We'll see how the choices look. So instead of having this fragment, this fragment, this fragment, now instead of cutting at these spots, you would have one big long fragment that would get stuck earlier on in your actual gel electrophoresis process, right? It's going to be longer. So instead of it looking like, let me undo that. Instead of it looking like this, you might have just one big fat one higher up. All right, so will the mutation create a new recognition site? It shouldn't be a new recognition site. It's mutating an existing one. The mutation would disrupt the restriction enzyme's ability to cut, leading to the absence of the expected fragments. That's sounding like what we have here. The mutation would have no effect on the enzyme activity. Well, you were telling me you're going to change where it cuts at. Of course, it's going to have an effect on it. The mutation would enhance it. No. It's going to inhibit it from cutting at places. So we'll have a different banding pattern. That's what's going to happen. Our next one is dealing with PCR. And again, our steps: denature, anneal, extend, or elongate, same thing. So I'm going to write those out here for PCR, denature, anneal, and elongate. See if this helps. Like I said, I don't reread the questions before I do this, so it'll be a surprise for me too. Which of the following best describes the purpose of the annealing step in PCR? Would this be to separate the double-stranded DNA into the single strands? No, that's not the annealing step, right? That's the denature step. To allow primers to bind to the complementary sequences, that is what the annealing step is for, but we'll read our other choices. Is the annealing step for synthesizing new DNA? No. Or is it to amplify the DNA by increasing the temperature? No. The anneal step is for binding the primers. It's B. A clinical laboratory is using PCR to detect the presence of a specific pathogen in a patient's sample. If the test is successful, what outcome would indicate a positive result? If the patient has the disease, then they should make products. That's pretty much how PCR works. So A, the PCR reaction produces no products. That would be a negative result. The PCR reaction yields a single large fragment that is larger than expected. No, you're making exact copies of what's already there. The PCR reaction generates multiple DNA fragments of the expected size. Yes, you'll be doing it over and over again, making many copies, indicating successful amplification. Yes. The PCR reaction results in the formation of a new type of primer. No, we're just making copies of what is already there and just making a lot of them. All right. I think we have one question for chapter 9. So this is dealing with the tonicity stuff. Who is surprised? Isotonic versus hypotonic versus hypertonic. Iso, remember that's going to be equal. Hypo, lower outside. So water moves in. This can burst a cell open, by the way, if it swells up enough. In the hyper, we have a higher salts or whatever it is, higher on the outside. So water moves out of the cell. And this is causing shrinking of the cell, which is also known as plasmolysis. Okay, so question 1, which of the following best describes the movement of water in an isotonic solution? We are saying that one is equal. Water moves into the cell, causing it to swell. Water moves out of the cell, causing it to shrink. Water moves in and out at equal rates, or water does not move. It moves in and out at equal rates. Okay, it's always moving. It's just gonna be at equal rates. No change in the net cell volume. Next question, a patient is receiving IV fluids and the physician orders a saline solution to be administered. The saline solution is labeled as 0.9% sodium chloride. Which type of tonicity is this solution and why is it chosen for the patient? In order to actually make sure that our patient should be hydrated as what we are expecting, this should be an isotonic solution. It maintains the balance of fluids between the inside and outside of the cells. We're not causing cells to shrink. We shouldn't be causing them to draw in too much water or release too much water. Only in very extreme cases will you be reaching for something that's hypo or hypertonic, very rare cases, and having to deal with kidney disorders and things like that. Normally, if you're reaching for saline to hydrate your patient, just make sure they're hydrated and there's no other concerns with how their body's dealing with that, then you're typically going to be reaching for isotonic, what we would expect the body to be, an isotonic. All right. Chapter 25, the effect of global warming on disease spread. And so this was leading more into like mosquitoes and stuff like that. So global warming, how does it contribute to the increased risk of mosquito-borne diseases? Mosquitoes are moving north, and so are the diseases that they carry, right? So does it decrease the breeding sites due to less rainfall? It's not that. Is it reducing mosquito populations by lowering? No. Is it expanding mosquito habitats and accelerating the development of mosquitoes in the pathogens they carry? Yes. Is it preventing replication of pathogens? No. So it's C. Which of the following public health strategies is most likely to be affected by the expansion of mosquito habitats due to global warming? Is it going to reduce antibiotic prescriptions to prevent antibiotic resistance? No. Is it going to enhance vaccination programs for mosquito-borne diseases in newly affected regions? Yes. If we have existing vaccines for that, we'll see that spreading to newly affected regions. We have promoting low-fat diets. That might help your health overall if you are morbidly obese. That could increase your health overall, but that's probably not going to be specifically related to the mosquito population increase. Will be because of increasing the use of personal protective equipment in non-endemic areas. Might that be an issue? That might be, but is that necessarily a cause of the mosquito population itself and how we react directly to that? B is our best answer. Finally, into chapter 10, I think we have a couple questions for it and then we'll be done. The enzyme structures. So the holoenzyme, the apoenzyme, the coenzyme. So the whole functional enzyme, we call the holoenzyme. The protein part is called the apoenzyme. The part that it needs to function, like the iron in hemoglobin, non-protein helper part, we call that the cofactor. If it is organic, we call it the coenzyme. Okay, so this is what we know as a conjugated enzyme. Not all enzymes need all of this BS to work. If they don't, then we call them simple enzymes, and they're just the protein, and it just works. But if you need this extra help from your cofactor or your organic cofactor, which would be coenzyme, then your protein portion is the apoenzyme. And the part that binds your pizza-shaped substrate is going to be on your apoenzyme, and that site is called the active site. So we have it all in one place. Okay, first question. Which of the following enzyme components is directly responsible for the chemical reaction by binding to the substrate? We have it drawn out here. Where are we binding the substrate? At the active site, which is part of the apoenzyme, but the active site is the better answer for this. Next question, in the context of enzyme deficiencies leading to metabolic disorders, which component's absence or dysfunction is most likely to affect the enzyme activity and result in conditions like phenylketonuria? So enzyme deficiencies, where you don't have your enzyme acting on your substrate, right? So if we don't have the apoenzyme, that's the part I would be the most concerned about because that's the part that actually acts on the thing. That's where your mutation would be happening. As far as like you have phenylketonuria, this is a genetic issue. What do you get from your genetics? You get proteins, right? And so the part of your enzyme that is the protein is the apoenzyme. So that's probably where your dysregulation is going to be. That's kind of just reading into the fact that this is a protein related dysfunction. Yay, redox stuff. Okay. So we are going to write down here, Leo says GER, so we don't forget that. Lose electrons oxidized, gain electrons, and you're reduced. In a redox reaction, which term describes a process where a molecule gains electrons? Gain electron is reduced. Reduction. In the management of certain diseases such as cystic fibrosis, medications that impact oxidative stress are used. Which type of reaction is directly targeted by antioxidants in these treatments? Okay, I'm starting to jump ahead with it because I feel like it gives it away in the question, but if it's oxidative stress, If there's oxidation, anytime there's oxidation, they are always together. If there is oxidation, you must have reduction. And so we're looking here at redox reactions. All right. Electron transport chain. In eukaryotes, the electron transport chain is on the inner membrane of mitochondria called the cristae. In bacteria, this is a whole ass bacteria right here. All right. This is a bacteria cell, and it might have a cell wall on the outside. Let's give them a wall on the outside. This is our gram positive. Okay. Got a nice thick wall on the outside. In bacteria, the electron transport chain happens on the inner membrane. Remember that electron transport chain is all those cytochromes, and we'll have the ATP synthase and everything. So in eukaryotes, this happens in the mitochondria in that larger cell that has a nucleus with DNA in it, whatever. And so this is your mitochondria. Okay. Where does the electron transport chain operate in bacteria? Is it on the inner mitochondrial membrane? Bacteria don't have a mitochondria. Is it on the outer mitochondrial membrane? I already said it. Cytoplasmic or plasma membrane. This is their inner membrane. Their inner membrane is also known as the cytoplasmic or plasma membrane. It's what contains their cytoplasm. All right. Is it their nuclear membrane? They ain't got a nucleus either. This question, another one that would have answered it for us. All right. In the treatment of mitochondrial disorders such as Leber's hereditary optic neuropathy, which part of the cell is directly targeted by therapies aiming at improving cellular respiration? We already know cellular respiration. If you're dealing with a optic neuropathy, this is in a eukaryote. And we already know that this is going to be in the mitochondria. That's where cellular respiration is primarily occurring, on the inner mitochondrial membrane. So it's not the cytoplasmic or plasma membrane. It's not the ER, and it's not the Golgi apparatus. It's our mitochondrial membrane. Last one. Oh, this is the last one, right? Yes. Okay. Like, please. Even me, man. It's a lot. So, all right. The generation of ATP. This is our proton motive force. And so this would have me answering these questions. We can revert back to that image that we drew of this thing. This up here is our proton motive force. We're creating those protons by exciting the cytochromes with our electrons, pumping protons out. We brought the protons and the electrons on our NADH and FADH2, just to review. We made those from glycolysis and Krebs, brought them here. They dropped off the electrons and the protons that they came with. The electrons excite the cytochromes. The protons get pushed out. We create the proton motive force gradient. They move back in through ATP synthase and drive the manufacturing of ATP. So what is the primary role of the proton motive force? It is to create that gradient that drives ATP synthase, right? So that's B. In the treatment of mitochondrial diseases, another mitochondrial disease, such as mitochondrial myopathy, so muscle issue, which cellular process is directly impacted by therapies aiming at enhancing the proton motive force? The proton motive force is essential in ATP production in our electron transport chain. So that's going to be C. Any questions that you have anyways? So you guys know, whether it's you or anybody else that watches this later on, so you guys know you can always email me if you have questions when you're studying and we're trying to figure this stuff out and make sense of your learning objectives. Otherwise, that's it for today. Thanks. That was a long one.